Buon pomeriggio ai presenti. Il tema di questo nostro incontro non è scritto, mi pare, sullo schermo, ma era una svolta, l'impulso per una svolta della ricerca di Biogem. In realtà la svolta si sta realizzando con questa progressiva internazionalizzazione di Biogem. Io devo esprimere un plauso particolare al direttore scientifico, il professore Capasso, che in una concentrazione di tempo molto breve ha creato delle grandi occasioni a Biogem di affacciarsi sulla scena internazionale. C'è stato il tema, il convegno precedente su rene cervello, c'è stato questo recentissimo che si è svolto a Napoli la settimana scorsa, è finito sabato, partecipato anche la nostra dottoressa Bianchino, oltre naturalmente alla parte, diciamo, il professore Trepiccione, insomma, la parte, diciamo, della nefrologia. Sono grandi occasioni per Biogem. Io devo esprimere anche con molta franchezza, lo dirò, lo dico qui, insomma, il rammarico di una presenza non adeguata a questo appuntamento. Rappresenterò questo ai responsabili dei laboratori. Sono occasioni preziose. Voi sapete che io insisto sempre molto sul fatto che noi siamo periferici, ma proprio per questo abbiamo una ragione in più di cogliere le occasioni per evitare la stagnazione, per evitare, diciamo, la mortagora che si può realizzare in una condizione, diciamo, di non stimolazione, come può avvenire in centri dove c'è un insieme di circostanze e di condizioni che questo favoriscono. Noi abbiamo il dovere di essere più attenti a questa questione. E questo l'ho voluto fare in pubblico perché anche se ci sono ospiti, ma insomma per sottolineare un punto negativo, avete espresso giudizi positivi su Biogem, io vi ringrazio, ma insomma bisogna che tutti siamo sempre sulla corda. Nessuno deve immaginare che insomma, ci si può cullare sugli allori, che durano, sono sempre effimeri. Quindi io non devo dire altro se non esprimere il benvenuto ai tre ospiti importanti che sono in prima fila. E la professoressa è professoressa che ha una radice italiana, anche se ormai è stabilmente americanizzata, ma noi confidiamo che le vicende possano realizzare anche un ritorno, perché insomma noi non solo per ragioni, come dire, un po' patriotticamente nostalgici, ma proprio nell'interesse dello sviluppo scientifico, non possiamo rassegnarci alla perdita di cervelli. Insomma, un saluto, noi ci siamo già scambiati saluti privatamente, prima nel mio studio, poi abbiamo avuto la possibilità di stare insieme a mensa, quindi non mi resta che rinnovare pubblicamente il ringraziamento per la vostra presenza qui e per il contributo che offrirete ai nostri giovani ricercatori, e con il rammarico che i non presenti perdono un'occasione preziosa. Grazie ancora e la parola al professore Capasso.
Thank you, Professor Zakino, President of the Biogem. And I would like just to tell you why we have worked for this special afternoon. We are, first of all, a research center. Yeah, research center in molecular biology and genetics, but in the last few years, we have also changed our attitude in order to be a research center in translational medicine. And this is a very competitive subject, where competition in research. Research means, first of all, meritocracy, not democracy, meritocracy, because that is the main difference with the political and the research. Research institute, any one of us can do research, but in order to be at the top level, she needs to be the best. In order to be the best, there are several issues we can get this performance of this let's say topic or this target. And one is that to be updated. Updated means several things. Means updated means that when we are doing an experiment, we should do updated what is the best technology to use. When we think an experiment, we should know what the other people has done. Because not to repeat an experiment must be every day something new, something original, some with this the creativity. And if you are not updated, you cannot be creativity. And in order to be updated, when you read a paper, this is old. What's important, not the paper when it's published, but the paper when it's sold. There is no other way to think what the paper will be published. That's to hear from different parts of the world the person that are doing experiment, they're creating new adventures, new trajectory, new theory. And in order to do that, it means just to organize time to time some talk, seminars, afternoon like this one. And just an indication of the previous meeting that we had. I have select 3 of the speakers that we had last week in Naples. And this speaker, I asked them, well, you are coming to Biogem for the first time. Please don't tell us what we have done in the past. Don't tell us what I'm doing right now. Just tell us what you will do tomorrow. And it's not dependent from what Biogem is doing, but what you are thinking, because when it is a new idea, the contamination of the ideas, this idea, is the paramount, is the fuel, or it is of a paramount important for our action of today. So they accept, and thank you very much. On the 3 of them, Farad, Alessia, and Rafael, we are becoming a friend. We have really friends in the past, but we are becoming more friends now. And they came to Biogem just to tell us, to the Biogem community. And the community means a family, and the family at Christmas time, at Easter time, we are all together. No? And today is a Christmas time for Biogem. Presidente, today, oggi è come Natale per Biogem. Okay, so that's and they tell us what they want, what they are going to do. They ask me, please, Gianni, please send to me each one what you are doing in Biogem, and we can try to propose a topic where potentially we will collaborate. But collaboration for them means also selection. They are one of the best research in the world in their own field. And they have to select and collaborate. And if we don't demonstrate that we would like to do something, of course they will not select to collaborate with us. So that is the target of the team meeting. First of all, to hear them, and then to show them, both to Alessia, Rafael, and Annette, that probably we can become competitive partners with them. So it's a dot to S, we can say in Latin. Noi vi diamo qualcosa per ricevere qualcosa. And hope at the end of today, just one afternoon, you will have impression that Biogem can be your partners in your research, and we can be your partners in your research, because that's the way, Mr. President, how Biogem can, grow and look to the future. Also, when we not be here, when we'll be in another place. That's the message that I want to give to young investigators. In nothing in the world you receive for nothing. You have to work for that. And work means that for a researcher, there is no Christmas time, there is no Easter time, there is no vacation, because mind, the originality, they don't take vacation. Michele is this. He works every day, 24 hours a day, and is happy to work 24 hours a day. He should be happy to work 24 hours a day. That's a message that we want to give it to you. Now I leave the podium to Michele for the introduction to the first speaker.
Okay, it's my pleasure to introduce this very important person in the research on lipid research and the connection between the lipids and the kidney. And Professor Alessia Fornoni is a tenured professor of medicine and molecular and cellular pharmacology at the University of Miami Miller School of Medicine. And she serves as director of the Peggy and Harold Katz Drug Discovery Center. And as a physician scientist who has maintained a resolutely focused research program, she gave a prominent contribution for the understanding of the pathogenesis of kidney diseases. And her research is supported by grants from National Institute of Health. She worked firstly at Roche in Basel. There is something in common with me because also my father worked for Roche, but in the United States as a chemist. And she is now in Florida at the Academy of Science, and she received several grants from national and private agencies in the United States, and she is also a reviewer for research grants for private and public agencies, and she is also in the editorial board of very important journals such as Journal of Clinical Investigation and Kidney International, and she has published several papers on very important journals such as JCI, New England Journal of Medicine, Nature Medicines and Metabolism, and so on. And she is pioneering in the study of insulin signaling, cholesterol metabolism, and sphingolipid-related pathways. So, Dr. Fornoni, please, Professor Fornoni, for your presentation.
Grazie per l'introduzione, sono molto contenta di essere qui. Abbiamo sentito il professor D'Acchino che ha detto non possiamo sederci sugli allori. Abbiamo sentito il professor Capasso che ci ha detto la meritocrazia è importante. E questo è importante perché se voi guardate questa slide, questo è Malpighi, che ha scoperto il glomerulo. Io sono nefrologa, vivo nel rene, e nel rene è l'unità filtrante il glomerulo. E queste sono virtue e fame, la virtù e la fama, che danno il dono a Malpighi per la scoperta. Per cui io vi auguro tutti di diventare voi i vostri Malpighi del futuro, insomma questo è davvero molto importante. Ho cercato di pensare un po' di cosa possiamo parlare e ho cercato di coprire un paio di topic dove potremmo trovare delle linee in collaborazione. Questa è la mia disclosure slide perché sono convinta che bisogna lavorare con l'industria e col privato per riuscire ad avanzare le nostre scoperte. Parleremo un pochino delle collagenopatie, poi dei meccanismi di lipotossicità di cui mi occupo da 15-20 anni, parleremo un pochino di come queste scoperte hanno portato allo sviluppo di un farmaco e poi parleremo di alcuni target. E la nefrologia è un campo molto eccitante, qua c'è molta oncologia, c'è molta nefrologia, ma in realtà mentre a nefrologia non facciamo mai farmaci e fare il nefrologo diventava quasi un'attività un po' noiosa. Ultimamente stiamo scoprendo un sacco di nuovi farmaci, ne abbiamo parlato a pranzo con alcuni di voi e questo ci rende molto competitivi anche col mondo dell'oncologia. E una delle patologie di cui mi occupo io è Alport syndrome. Questa è una patologia che era pensata come una patologia ultra rara dovuta a mutazione delle catene alfa del collagene di tipo quarto. E nella Alport Faccio colmo. Nella Alport si mutano o la catena quinta che è sul cromosomo X o sulla catena terza e quarta che sono su altri cromosomi. Però con l'avvento della genetica si è scoperto che in realtà se guardiamo la popolazione di pazienti che hanno malattia renale cronica, c'è una quantità di pazienti abbastanza alta che ha queste mutazioni. Nessuno li ha mai chiamati Alport perché non abbiamo mai fatto gli studi genetici. Ma adesso che facciamo gli studi genetici quasi in tutti, abbiamo visto che più o meno il 30% dei pazienti che hanno il danno renale cronico hanno mutazioni in queste catene del collagene, che davvero fa pensare che questa malattia non sia poi così rara. Persino quando si fanno studi di genetica sul diabete si è visto che alcune varianti, SNP variant in questo caso, del collagene, delle catene alfa del collagene, correlano con il danno renale. Per capire però una cosa che non abbiamo ancora capito qual è la correlazione tra il genotipo e il fenotipo in queste collagenopatie. Questa è rimasta una cosa abbastanza difficile e una delle ragazze a cui faccio mentoring, vi faccio vedere un paio di dati, ha sviluppato un essay che può aiutarci a capire questa genotipo-fenotipo correlazione. Non voglio perdere troppo tempo, ma questo è come il collagene tipo quarto. C'è un N-terminal domain che ha parecchie cisteine, come pure c'è un globule C-terminal NC1 domain da questa parte, mentre qui è la parte dove c'è la trimerizzazione. E mutazioni da una parte o dall'altra del collagene tipo quarto possono influenzare il tipo di patologia. Quello che non sappiamo è esattamente qual è questa correlazione tra genotipo e fenotipo e l'interpretazione spesso come nefrologa, quando chiediamo il genotipo dei pazienti ci arriva questo report che dice variant of unknown significance e noi non sappiamo cosa fare. Abbiamo davanti un paziente ammalato, la genetica ci dice negativa perché è mai stata riportata questa variante, cosa ne facciamo di queste varianti che ci ridà la genetica? E allora sono stati sviluppati dei test come questo test che si chiama split luciferase, dove praticamente il segnale di luciferasi viene emesso quando la catena di collagene tipo quarto si può fare in modo trimerico come si dovrebbe fare. E non solo si può vedere se si assembla la catena del collagene, ma si può vedere se viene secreta, se rimane interna alla cellula, se fa quello che deve fare. E delle diverse varianti che sono stati descritti ai pazienti si può vedere, come dicevo, quando c'è un difetto di trimerizzazione non viene fatta luciferasi, quando c'è un difetto di secrezione la luciferasi rimane interna alla cellula e quando invece la genetica non condiziona né la secrezione né la produzione del collagene di quarto, il collagene può essere prodotto. Jennifer E., che lavora con me da Ann Arbor, Michigan, ha sviluppato questo sistema per studiare tutte le mutazioni che noi riconosciamo. Io sono il PI a livello nazionale negli Stati Uniti di uno studio di natural history del paziente con l'Alport e collezioniamo DNA, collezioniamo siero, collezioniamo urine, il goal è di avere 100 pazienti, siamo a 60 adesso oltre a questo sistema di genotipo, di questo split luciferase che vi ho fatto vedere, sarebbe molto bello poter lavorare insieme a fare degli zebrafish che hanno le diverse mutazioni che noi riconosciamo dei pazienti per vedere se abbiamo una correlazione tra il danno che vediamo nel zebrafish e quello che vediamo nel paziente e per fare dei test predittivi per sapere se il zebrafish ha la proteinuria o no. Questa è una delle possibilità. Passo alla seconda parte del talk, dove vi voglio far vedere un pochino quello che è stata la mia ricerca negli ultimi anni. Come dicevo prima, io vivo nel glomerulo, che è la filtrazione, il sistema di filtrazione del rene, e nel glomerulo vivo soprattutto con questa cellula che si chiama podocita, che sta sul lato urinario della filtrazione glomerulare. Però spesso e volentieri non sappiamo cosa porta questa bellissima cellula con questa struttura architettonica molto bella a una cellula piatta che non ha i pedicelli podocitari e finché non sappiamo cos'è che porta dal sano al malato non sapremo come riportarlo. Una cosa che è chiara è che in parecchie patologie renali c'è un danno del podocita, che noi guardiamo all'Alport, al diabete, all'FSGS, che siano patologie metaboliche o non metaboliche, il podocita viene sempre, si ammala. Non solo si ammala, ma è sufficiente che si ammali il podocita per avere un'insufficienza renale cronica. È stato spiegato, è stato dimostrato con metodi sperimentali e clinici che la riduzione del numero di podociti, che è una cellula che non può proliferare, è sufficiente a causare il danno renale cronico. E in realtà che guardiamo a un Alport o a un paziente con diabete, c'è questa alterazione della morfologia del podocita che si siede sulla membrana basale glomerulare. Una delle teorie è quella che quando la membrana basale è alterata, il podocita in qualche modo cade, come possiamo cadere noi più facilmente quando camminiamo su una superficie non liscia. E come pure però quando sappiamo che dobbiamo camminare sulla superficie non liscia ci mettiamo delle scarpe più comode. Il podocita indossa questa scarpa di avere una risposta metabolica, si prepara, accumula droplet lipidiche, preparandosi alla possibilità di cadere e di aver bisogno di energia. In modo abbastanza semplistico. Abbiamo dimostrato che se isoliamo cellule dal paziente diabetico, dal paziente con Alport rispetto al controllo, accumulano qui in rosso nei droplet lipidici e come qui in verde nell'Alport. Perché sia che la patologia sia metabolica che non metabolica c'è l'accumulo di droplet lipidiche. E non c'è dubbio che il lipide abbia un'importanza fisiologica nel rene, perché se noi guardiamo al filtro renale, che è costituito da tutte queste molecole che interagiscono tra di loro, le molecole possono interagire solo se c'è una composizione giusta di colesterolo, fosfolipidi, sfingolipidi, di quelli che chiamiamo lipid raft domain, a livello del filtrato glomerulare. Ma i lipidi, cosa succede se si accumula troppi lipidi nel tessuto? E l'accumulo di lipidi nel tessuto è stato descritto nei pazienti con nefropatia diabetica. Queste sono droplet lipidiche nel podocita di un paziente con nefropatia diabetica. Lo possiamo vedere nei dati sperimentali. Possiamo vedere che l'accumulo di colesterolo nel rene correla con i dati di funzionalità renale e questo succede anche in modelli non metabolici come Alport, che è un modello animale, un collagen 4 alpha 3 knockout mouse che accumula questo colesterolo nel parenchima, che fa pensare all'esistenza di un fatty kidney disease, no? E qui sono le droplette lipidiche e i cristalli di colesterolo nel tessuto. Quando guardiamo l'ipotossicità in Alport, è una cosa che inizia nei glomeruli juxtamedullari e poi va a colpire anche i glomeruli più periferici, poi le cellule tubolari e poi queste interstitial foam cells che si vedono soprattutto nella patologia da Alport più che di altre malattie glomerulari. E quando guardiamo la dropletta lipidica, per me è diventata proprio un organello molto importante. Queste sono due immagini del laboratorio che ci fanno vedere che in condizioni normali le droplette lipidiche ci sono e che non hanno nessun problema ad avere il contatto con il mitocondrio, per cui a servire come sorgente di energia al mitocondrio. Ma in danno patologico, Alport, ma anche altre forme di malattia renale cronica, si accumulano più droplette lipidiche, non solo si accumulano ma non possono essere utilizzate, non le vediamo mai attaccarsi ai mitocondri. E questo mi porta a parlarvi un po' della scienza. Inizialmente abbiamo pensato che questo accumulo di lipidi fosse legato a un aumento dell'uptake di questi lipidi. E un'ipotesi che è stata testata a Jinju, una junior faculty che lavora con me, è che quel collagene tipo 1, che oggi abbiamo parlato un pochino di questo senescence markers, di questo collageno che viene accumulato in modo un po' non organizzato nei tessuti che invecchiano, Questo accumulo di collagene tipo 1 si vede anche nel paziente dell'Alport che non può fare il collagene tipo quarto, e quando si fa tanto collagene tipo 1 abbiamo attestato l'ipotesi che questo collagene fosse responsabile dell'accumulo di droplet lipidice, e Jinju ha dimostrato che esporre i podociti al collagene tipo 1 aumenta la dropletta lipidica e lo fa tramite un sistema di attivazione di un tyrosine kinase receptor che si chiama Discodin Domain Receptor 1, DDR1. Più collagene, più attivazione del DDR1, più accumulo di droplette lipidici con l'uptake del fatty acid. E cosa che ci ha fatto pensare, come abbiamo rivisto recentemente in questo Nature Review che abbiamo scritto, che il danno renale, che l'accumulo di dropletta lipidica possa essere legata a un uptake del lipide, soprattutto nella forma di fatty acid. Però no, a quanto pare non è l'unico sistema, perché se noi blocchiamo alcuni dei sistemi di fatty acid uptake, la patologia, l'accumulo di lipidi occorre comunque. Per cui abbiamo dovuto studiare altri meccanismi. Ci siamo chiesti se fosse una diminuita utilizzazione di queste lipid droplets, e questo è un lavoro dove abbiamo passato 4 anni per poi scoprire che stavamo in realtà seguendo un fantasma, e qui dove chiedo aiuto. Abbiamo pensato che questo potrebbe essere dovuto alla mancanza di queste proteine che si siedono sulla superficie della dropletta lipidica, come la Clean5, che aiuta la dropletta lipidica a contattarsi al mitocondrio. Che è ridotto nei pazienti con Alport a livello glomerulare, ci sembrava fosse ridotto anche nei glomeruli del topo con l'Alport rispetto al wild type, e abbiamo visto che se noi riesprimiamo Plin5 nel podocita di Alport possiamo riattaccare le droplette lipidiche al mitocondrio, possiamo utilizzarle come fuel. Abbiamo poi scoperto che l'esetimib, che è un farmaco che viene utilizzato spesso per l'iperlipidemia, può aumentare l'espressione di Plin5, e utilizzando l'esetimib abbiamo riattaccato alcune droplette lipidiche al mitocondrio, abbiamo migliorato il quadro renale. In effetti l'esetimib riduce l'albuminuria, riduce il danno renale alla creatinina, migliora il danno istologico e riduce la quantità di trigliceridi in rene senza però cambiare la quantità di colesterolo. Questo ezetimibe è stato adesso studiato anche da un punto di vista clinico dal nostro collaboratore Peter Rossing a Copenaghen, che ha studiato alcuni pazienti con diabete tipo 2 facendo risonanza magnetica, dimostrando che l'esetimibe nel paziente albuminurico può ridurre la quantità di lipidi che si possono detettare con immagini, con risonanza magnetica del rene. Ma c'è un problema, abbiamo lavorato per 4 anni su questa molecola e abbiamo scoperto che non stiamo guardando la molecola giusta e non sappiamo cosa stiamo guardando. Perché vi dico questo? Abbiamo importato, c'è stato un momento in cui ho avuto il dubbio che questa molecola, che veniva riconosciuta da tanti complessi diversi, si vede con il messenger RNA, che davvero non fosse Clean5. Ci siamo fatti mandare dei pezzi di tessuto di Clean5 knockout mice e l'anticorpo funzionava lo stesso. Per cui sappiamo purtroppo 4 anni dopo che questo Clean5 non esiste o non è il mediatore. E qui davvero dove c'è un'altra opportunità di collaborazione, ho visto delle mass spec bellissime questa mattina a Biogem e non faccio fatica a capire, vorrei davvero capire cosa riconosce questo anticorpo che abbiamo utilizzato, che viene venduto come anticorpo per Plin5 e che dà questo western blot che davvero sembra bianco e nero, no? Il controllo verso il topo con CKD, questa banda scompare. Abbiamo provato a fare una mass spec, non sappiamo cosa sia, Plin5 non esce su questa banda, e se qualcuno di voi viene voglia di avere la challenge di capire che cos'è che riconosciamo, io penso che questa potrebbe essere una molecola molto importante da studiare, perché come ben sapete siamo stati generosi a chiamarla ricerca e non ricerca. No? La facciamo così tante volte e per me questo finding però bianco e nero su questa proteina mi interessa molto e vorrei davvero sapere di che cosa si trattasse. un altro meccanismo per eliminare queste droplette lipidiche è cercare di aumentare il loro cioè pensare che probabilmente non riusciamo a eliminarle in modo appropriato. E questo è un po' quello che è stato studiato nell'aterosclerosi, dove quando si forma il macrofago foamy macrophage nella placca aterosclerotica, questo è dovuto a un'incapacità di fare cholesterol efflux della cellula alla HDL. E ci siamo chiesti se questi meccanismi che sono responsabili per formare il macrofago lipidico nell'aterosclerosi fossero presenti anche nella microvascolatura del rene. Ispirati dal fatto che ci siano due malattie renali genetiche nell'uomo, Tangier disease e Elcat deficiency, queste due malattie sono caratterizzate dall'accumulo di lipidi nel rene e sono accompagnate dalla proteinuria. Non c'è niente come la genetica umana che può suggerire un effetto di causa-effetto tra il danno renale e il fenotipo. La prima cosa che abbiamo fatto è ci siamo chiesti se i podociti esprimessero tutti i fattori che danno la possibilità di fare il metabolismo lipidico. I podociti possono prendere il colesterolo con l'LDL receptor, lo possono sintetizzare con le CMG con i reductase, lo possono mandare all'HDL, esprimono tutti i fattori che sono responsabili per il colesterolo. Ma la prima cosa che abbiamo fatto, e questo vi do 5 minuti per dirvi quello che abbiamo fatto negli ultimi 10 anni, abbiamo scoperto che in realtà il colesterolo si accumulava nel podocita del paziente con la nefropatia diabetica perché non riusciva, il podocita non esprimeva abbastanza di questa molecola ABC1 che è il donatore di colesterolo alla HDL. E abbiamo visto che sia nelle biopsie renali dei pazienti che in podociti in cultura trattati col siero del paziente diabetico scompare l'espressione di questo trasportatore del colesterolo. La stessa cosa succede in un modello di Alport, per cui anche in un modello non metabolico. Abbiamo scoperto e pubblicato su JCI che quello che guida la soppressione di questo ABC1 è l'infiammazione locale, soprattutto il TNF-alfa che abbassa l'espressione di ABC1. E abbiamo scoperto che è sufficiente overesprimere ABC1 da un punto di vista genetico per curare il danno renale, per regressare la patologia renale e la proteinuria. Abbiamo scoperto anche che non avere l'ABCA1 a livello podocitario non è sufficiente per fare la patologia, e questo è stato il lavoro di Michel Ducasa, che però ha anche scoperto che il mitocondrio non funziona bene quando ci sono troppe droplette lipidiche. E voglio spostarmi adesso a parlare un attimino a come queste ricerche che ci hanno fatto vedere che l'efflusso di colesterolo è importante per la patologia renale. Ci siamo chiesto, ok, se riusciamo a esprimere una molecola geneticamente che cura la malattia renale, possiamo fare un farmaco o no? E questo è stato quello che a un certo punto della mia carriera mi ha fatto fare questo salto dall'accademia alla industry, perché mi sembrava di perdermi in questa loop dove facevo più pubblicazioni, più fondi, più dati, però io volevo portare le mie scoperte alla comunità. Allora ho negoziato un paio di anni con l'accademia, ho fatto questo salto a quella che io chiamo la Valley of Death, scusate, la Valley of Death in Translational Medicine, e sono andata con le mie scoperte dall'Accademia a Basilea in Svizzera, dove sono riuscita a capire come si può scoprire un farmaco una volta che abbiamo un target sicuro in mano. E qui in collaborazione con Marco Prodotto e Matthew Wright abbiamo praticamente sviluppato un farmaco, una small molecule, un 5-arenine nicotinamide compound, che riesce a causare l'efflusso di colesterolo tramite l'effetto su questa molecola di cui vi ho parlato, che è l'ABCA1. E questo farmaco, dato ad esempio in un animale sperimentale con l'Alport, riduce drammaticamente l'albuminuria, la creatinina, tutti i parametri funzionali renali migliorano in modo drammatico e questo è un esemplare istologico dove potete vedere che i glomeruli ritornano più o meno normali e anche quando il farmaco viene dato a una fase in cui la malattia è relativamente avanzata, i topi sopravvivono più a lungo e questi sono topi che muoiono di insufficienza renale. Molto interessante. Questo farmaco riduce anche la quantità di droplette lipidiche nel glomeruli del topo di Alport e vi posso dire questo farmaco è poi stato testato in trial clinici fase 1, non ha avuto nessun problema e adesso sta reclutando a livello mondiale in fase 2 per la malattia di Alport e altre malattie proteinuriche. Per cui potete immaginare l'eccitazione che come physician scientist uno può avere a fare una scoperta che inizia al paziente, arriva in laboratorio, si fa la cella, si fa il topo e adesso si fa il farmaco che torna ai pazienti. Il mio sogno è di ridare alla comunità di pazienti di cui tratto e spero che succederà. Questo farmaco non si lega direttamente all'EBCA1, questo farmaco si lega a una proteina che si chiama oxysterol binding protein 7, che è una proteina che serve per fare il traffico di lipidi tra organelli e organelli. La stiamo studiando, non sappiamo cosa sia, non c'è stato quasi niente scoperto su questa molecola prima. Jeff Presley, che è un postdoc nel laboratorio, ha sviluppato un zebrafish model con Mario Schiffer, dove non c'è l'OSBPL7 e può vedere che c'è più proteinuria e più sindrome nefrosica. È molto difficile per noi continuare collaborazioni a distanza, per cui speriamo che qualcuno di voi venga voglia di lavorare con noi sul zebrafish model che vogliamo sviluppare e io sarei molto contenta di fare il mentor scientifico, ma lasciare il progetto con voi per riuscire a capire altre cose di questa molecola. Per cui oltre a utilizzare questo modello zebrafish, come vi ho detto prima, per studiare le diverse patologie di Alport, così una possibilità sarebbe quella di anche sviluppare un topo. Non esiste un flux topo, un topo che è flux tag o che è un inducible model for SBPL7 e so che voi avete una facility per la generazione di transgeniche knockout e questa potrebbe essere una possibilità, una cosa che io non ho ancora iniziato ma che vorrei fare. Veramente 2 minuti per non rubare troppo tempo ai miei colleghi. Continuiamo a lavorare anche su nuovi target, no? Abbiamo sviluppato questa molecola, è una small molecule, ma vogliamo fare anche altre cose. Un'altra cosa che abbiamo scoperto, e so che voi siete forti anche a sviluppare i biologici, è scoprire che nella malattia renale cronica c'è una deficienza renale di una proteina che si pensava fosse prodotta solo dal fegato, la apolipoprotein M. Questa apolipoproteina fa parte anche quella della HDL, è anche un'apolipoproteina che è responsabile del flusso di colesterolo. E quello che abbiamo visto dando una apolipoproteina ricombinante legata alla frazione Fc di un anticorpo è che, no scusate, innanzitutto abbiamo visto che potrebbe servire da biomarkers, anche se purtroppo non abbiamo un ELISA molto buono per studiare questa proteina. Un'altra cosa che potremmo fare insieme è chiaro studiando una patologia glomerulare di sindrome nefrosica che l'epolipoproteina M è bassa rispetto ai controlli. Abbiamo anche scoperto che se ridiamo il biologico, se ridiamo la proteina ricombinante, abbiamo una normalizzazione quasi completa dell'albuminuria e del danno renale. E abbiamo provato a fare un co-development agreement con Pfizer di questa molecola, ma Pfizer ha chiuso il campo delle malattie renali e ci troviamo ora a dover cercare nuovi partner da questo punto di vista. E questo potrebbe essere uno sbocco di co-sviluppo di un biologico che può applicabile alla patologia renale. Per cui questa è la mia quarta opportunità, quella di sviluppare un nuovo biologico e quella magari di sviluppare un ELISA che potrebbe essere clinical grade per fare un nuovo biomarker nella patologia renale. Queste sono solo alcune scelte delle tante cose che potremmo fare insieme. Questo è il gruppo con cui lavoro io, Miami è sempre estate, per cui sarebbe, vabbè, qui magari pure si sta meglio che al nord dell'Italia, però mi piacerebbe un sacco potervi ospitare, poter avere uno scambio. O io imparare da voi, siamo tutti, un'altra cosa che diceva il professor Capasso è l'importanza del teamwork, no? Non si lavora insieme nella ricerca, in ricerca possiamo scoprire, avanzare, accelerare solo se lavoriamo tutti insieme, per cui non c'è gerarchia, c'è la conoscenza di ciascuno di voi e insieme ce la possiamo fare. E voglio finire qua dicendo sono proprio felice di essere qui e spero di comunicarvi un po' di passione. Io ce l'ho e mi sento ancora pagata per il mio hobby preferito, che è quello che fare il mio lavoro. Per cui vi auguro la stessa cosa. Non è impossibile toccare le stelle se volete toccare le stelle. E finisco qui invitandovi al congresso di Buenos Aires, che è il congresso internazionale di nefrologia. Io sono, quest'anno lo dirigo io, sono direttore scientifico e abbiamo bisogno di più menti giovani per innovare. Grazie mille.
Grazie molte per questa presentazione, veramente molto, molto bella. Non so se parla in inglese o in italiano. In italiano. Ovviamente vorrei rubare un paio di domande prima alla platea. La prima è veramente molto interessante quei dati sulle collagenopatie. Quindi il 30% dei pazienti con insufficienza renale cronica praticamente hanno scritto nel loro DNA che devono sviluppare ovviamente l'insufficienza renale cronica. E voi avete messo a punto un kit con delle mutazioni specifiche per andare ad investigare questo tipo di mutazioni e se questo tipo di mutazioni possono, la scoperta ovviamente di avere questo tipo di mutazioni possono essere utili anche da un punto di vista terapeutico per i pazienti stessi.
Sì, grazie. L'unica cosa che abbiamo sviluppato per ora, e questo non è il mio lavoro, è il lavoro di Jennifer E., la collaboratrice a Michigan, è questo luciferase split assay, dove possiamo trasfettare cellule con le diversi variant of unknown significance che ci arrivano dai pazienti e le a seconda dell'intensità della luciferasi e la localizzazione luciferasi può intuire se quella mutazione è patogenetica o no. In realtà sarebbe un passo troppo grosso passare solo dalla luciferase al paziente e avere invece una zebrafish facility che sviluppa tutte queste variant of unknown significance sarebbe eccezionale.
E questo può essere utile dal punto di vista clinico per il paziente?
Sì, perché per quello sicuramente sarà utile perché quella variant of unknown significant potrebbe diventare una variant of known significance. L'obiettivo è quello di trasformare l'unknown to known e per cui avremo la capacità di influenzare le compagnie che fanno genetica nei pazienti e fare cambiare la nomenclatura della variante.
Da un punto di vista invece delle lipid droplets, voi avete visto che c'è un incremento del numero, ma anche qualitativamente questi lipid droplets sono diverse rispetto al...
Sì, questa è una domanda molto importante. Stiamo cercando di avere abbastanza risoluzione per vedere le dimensioni delle lipid droplets e la subcellular localization. L'unica cosa che riusciamo a vedere per ora è se sono attaccate ai mitocondri o no, ma sicuramente la dimensione della dropletta lipidica e la compartimentalizzazione dentro la cellula sarà molto importante in futuro. Non so ancora risponderle.
L'ezetimibe, no? Il meccanismo d'azione qual è?
Il meccanismo d'azione dell'ezetimibe dovrebbe essere solo quello di attaccarsi alla Niemann-Pick protein e prevenire l'assorbimento di colesterolo. In realtà tutti pensano che l'ezetimibe fosse solo un farmaco che entra ed esce e che previene l'assorbimento di colesterolo. ma ci sono stati poi lavori che hanno dimostrato che l'esetimide viene assorbito e che può alterare il metabolismo lipidico a livello del fegato, che è quello che ci ha fatto pensare che potrebbe anche funzionare a livello renale. Io in realtà non penso che si sappia esattamente, il Niemann-Pick-C1 è la proteina a cui si lega l'esetimide, ma penso che ce ne siano altre.
E le statine non potrebbero essere utili.
Le statine riducono la sintesi,
Sì.
no? Fanno il target dell'HNG con le reductase e noi non abbiamo mai dimostrato che la statina né previene il danno podocitario né previene la proteinuria. In realtà anche i dati clinici, noi le diamo le statine al paziente con insuccienza renale cronica per il danno cardiovascolare, ma non perché crediamo che rallentino il danno renale. Il problema non è la sintesi del colesterolo a livello renale, il problema è una combinazione tra una TEI con un'aumentata sintesi e l'incapacità di eliminarlo.
E per quanto riguarda gli sfingolipidi, invece ho visto che si occupano anche di sfingolipidi, quindi volevo sapere se c'erano delle novità, essendo diciamo un po' una mia fisima, diciamo.
Sì, non ho parlato di sfingolipidi per interesse di tempo, ma siamo molto, in realtà un grant che devo mandare via domani è un grant che studia l'effetto degli sfingolipidi attivi, sfingosin 1-phosphate. Tra l'altro abbiamo scoperto un enzima che cambia la ratio tra questi due sfingolipidi attivi, sfingosin 1-phosphate e ceramide 1-phosphate, che per me sono comunque molto molto interessanti. E abbiamo visto che se riusciamo a catturare l'S1P, riusciamo a prevenire il danno renale. Un meccanismo per catturarlo è questa apolipoproteina M, che è il substrato naturale per sfingosin 1-phosphate. Penso che ci sia abbastanza relazione e ho sempre pensato di lavorare in due campi diversi, sfingolipidi e colesterolo, ma adesso si stanno avvicinando sempre di più.
E come regolatori del traffico vescicolare di sfingolipidi, secondo me può avere un ruolo anche nell'accumulo poi ovviamente delle lipid droplets all'interno della cellula.
Sì, lo stiamo guardando adesso, sto cercando di capire adesso, non solo se ha un effetto diretto sull'accumulo dei lipidi all'interno della cellula, ma se può avere un effetto di prevenzione della dropletta lipidica nel seguire un autophagic flux. Penso che gli sfingolipid interferiscono più con l'autofagi o non autofagi l'ipofagi della dropletta lipidica.
Ok, veramente molto interessante. Ci
Grazie.
sono domande da parte dell'audience? Prego, Rossella.
Avevo una domanda in merito a cosa diceva che piuttosto che non mica una scelta. In merito a questo mi chiedevo se avete stato a fare diciamo un allineamento, cioè dipende da proteina, allineato rispetto alle proteine con una banca di vedere la similarità con le proteine così che restringe il campo di proteine da studiare.
Sì. Grazie, abbiamo fatto questa cosa e ci sono 6 plane. Abbiamo già escluso che si tratti di plane 1, plane 4 perché abbiamo avuto i knockout di questi animali e l'anticorpo non lo riconosce. Però siamo ancora di omologia ce n'è parecchia tra le diverse planes, per cui è un pochino difficile cercare di capire, non sono nemmeno più sicura che sia un plane, però qualsiasi cosa che vi posso dare informazioni sull'anticorpo che abbiamo usato e il plane 5 e se ci potete aiutare a capire perché, che cosa potrebbe essere questa molecola che scompare, che viene riconosciuta questo anticorpo, la compagnia dice esclusivamente riconosce il plane 5, non è vero. Non è vero e non sappiamo come andare avanti con questo progetto.
Prego, c'è un'altra domanda. Prego.
Grazie per la presentazione. Una domanda sull'utile clinico, che ha parlato di quanto sono grandi i numeri pulsanti della notazione delle proteine per il collagene, che non tipicamente poi non rientrano tutti in un quadro franco bianco. Io mi chiedo, delle molecole che avete studiato nell'ambito proprio della riduzione rimaneggiante delle proteine, che effetto possono avere sui pazienti invece che vanno in supplenza tonica, che sfruttano proteine che progrediscono ma che tipicamente sono lontani dall'altro? Cioè, se possono beneficiarne anche loro e se possono aumentare il numero di supportare poi studi per questo.
Sì. Grazie, abbiamo iniziato con l'Alport solo per una business decision perché come magari lei sa, i brevetti hanno 7 anni in più di vita quando si inizia la malattia renale, per cui questo spesso è una business decision che non dipende dallo scienziato che ha inventato. E in ogni caso adesso stiamo facendo questo trial sia sull'Alport, lo stiamo facendo sull'FSGS primary e lo stiamo facendo sulla nefropatia diabetica con Peter Rossing in Copenaghen, per cui speriamo. Io penso che la malattia renale cronica sia una forma di microaterosclerosi iniziata da fattori diversi, o genetici o metabolici, così, e che il farmaco dovrebbe riuscire a funzionare in tutti.
Prego, Lucia.
Sì, io ho 2 domande. La prima, non sono di questo campo, quindi magari sono banali, però la prima riguarda la regolazione col farmaco poi sul colesterolo, in particolare se le cellule target, tu hai parlato della diversa disposizione dei lipid droplets nella cellula, ma se le cellule target hanno una variazione metabolica, magari anche sotto trattamento con il farmaco, se sì quale, e se avete studiato se cambiano caratteristiche meccaniche, tipo la stiffness.
Sì, è bellissima questa domanda. Stiamo cercando di lavorare con l'Atomic Force Microscope dell'università per capire l'elasticità, anche se vorremmo capire l'elasticità degli organelli, non solo quella della membrana plasmatica. Per me non è stato facile. Se ci volete aiutare bene, per me non è stato facile. Allora, quella è la domanda numero. E il metabolismo abbiamo guardato, sappiamo che la dropletta lipidica rende il metabolismo meno efficiente. Ad esempio, pur avendo più cardiolipina e più super complex formations nel mitocondrio, c'è meno ATP production, per cui questa è una respirazione inefficiente e non sappiamo bene perché.
Pure su questo forse si potrebbe mettere.
Sì.
La seconda domanda in realtà era sul NPL5, eccetera, eccetera, ma io non so se l'anticorpo che voi avete utilizzato è sempre l'anticorpo che è stato mostrato dove si vedeva la regolazione anche farmacologica che aumentava, è sempre quello, quindi la proteina che aumentava non si sa che cos'è.
Sì, non sappiamo cosa sia.
Quindi forse anche studi, insomma, cercare di capire come viene regolata, studi di promotori degli NPL femmini.
Sì, forse sarebbe importante.
Di pigliarla così. Io pensavo a un isoforma più che altro, perché che sia proprio un familiare, uno della famiglia, allora questi che hanno fatto anticorpo devono cambiare mestiere. Però ho detto però forse un isoforma non ancora identificato, una cosa del genere. Penso che si potrebbe andare verso questa direzione.
Sì, ha ragione, sono carente io di conoscenze di come andare in questa direzione, ho bisogno, però sì.
Nel nostro piccolo, perché
No, bene, benissimo. L'unica cosa che posso dire è che quando abbiamo riespresso il Plin 5, pensando che fosse Plin 5, noi un pochino l'abbiamo restaurato quello fenotipo.
Quindi è qualcosa di simile.
Per cui è qualcosa di simile, è l'unica cosa che posso dire.
Subito ho pensato, non lo dico, non voglio fare una storia ora qui su aspetti post-traduzionali.
Può darsi.
Ma no, perché se il knockout...
Ah no, il knockout, sì, hai ragione, sì, ha ragione.
Per cui pensavo a un discorso di suo forma like.
Grazie.
Prego, Vincenzo.
Grazie mille per la presentazione. A me più altro è una curiosità che è nata da vari punti della discussione. Cioè mi chiedevo, specialmente per la regolazione delle droppole lipidiche, quale potrebbe essere il peso dell'epigenetica nella regolazione delle stesse? Partendo anche dal fatto, cioè io non ho potuto fare a meno di collegare il circuito a tutto un discorso.
Sì.
Well,
le sirtuine regolano il metabolismo lipidico, come pure però la dropletta lipidica regola l'HDAC. No? Per cui sicuramente c'è una correlazione di dropletta lipidica e epigenetica. Non l'ho mai esplorata perché non sono esperta di epigenetica.
Comunque è una modifica della nicotinamide,
Sì.
che è un inibitore delle sirtuine.
Sì, hai ragione.
Quindi di conseguenza io non ho fatto altro che pensare a questo.
Forse c'è l'aspetto così perché magari qualche sirtuina la sta già prendendo. Potrebbe essere.
Potrebbe essere.
Facile da verificare questo.
Potrebbe essere. Sì, no, abbiamo raggiunto un collaboratore all'istituzione che lavora sulle sirtuine per chiedere se ci fa un po' di blogs. Vorrei aprire questo campo, ma come sempre si ha un po' paura ad esplorare una parte nuova dove non abbiamo mai lavorato. Ma grazie, interessante come idea.
Ci sono altre domande? Sembra di no, quindi
Grazie.
Posso fare un commento?
Prego.
Alessia è venuto qui per diciamo, offrirci una collaborazione. Invece vedete come è molto piccolo, come parlare tra di noi in qualche modo si riescono a fare tante cose. Abbiamo qua adesso la proposta da meno di tre progetti da fare insieme che vengono da non esperti della materia. Lucia è un'oncologa, da altri due ancora,
Al futuro.
credo che ha trovato dei punti. Così come volevo
Io sono un
sottolineare.
fotologo generale.
Allora, volevo sottolineare anche Lucia, che l'importanza di quello che Alessia ci ha detto, perché ci sono stati in nefrologia non uno, ma tre grossi trial clinici che hanno in qualche modo cercato di capire se l'ipidemia avesse a che fare o no con l'insufficienza renale. Sono costati milioni di euro utilizzando le statine. Quindi abbiamo concluso che il colesterolo non ci azzecca proprio con l'insufficienza renale. Vedete un po', invece, gli studi che ha fatto lei, in cui ha dimostrato non dovete guardare alla colesterolemia, che poi è meglio un'altra cosa, ma dovete guardare al colesterolo intracellulare. Quindi lei ha aperto un campo completamente innovativo per la prevenzione delle malattie rare. Ma se il colesterolo è importante anche per il cuore, è importante anche per il cervello, vedete che non so quanto sia importante per la cellula aeroplastica, tanto per dire, ma vedete come ci si infetta in modo positivo ogni volta si apre, si permette di parlare a delle persone alle quali probabilmente dicono qualcosa di nuovo e quello che ha detto è una cosa di nuovo. Quindi noi accettiamo fondamentalmente quello che ci è chiesto di utilizzare lo 0,5, spero che ci sono gli esperti di tempo, cercare di avere un contatto con te in modo da scambiarci non solo idee ma progetti sperimentali.
Grazie.
Ok, grazie ancora. Complimenti. Passo alla seconda.
Il nome della presentazione? The name of the presentation?
So, in the meantime, it is my pleasure to
Naples.
introduce
Professor
Farah Danesh from the
Naples.
Division of Internal Medicine of the MD Anderson, Houston, Texas. And I would like to spend a few words. Actually, his research understand pathological and molecular basis of diabetic nephropathy, and many projects of his lab are on this. And the name of his presentation today will be micro and long non-coding RNAs in diabetic kidney disease. So, Farad, I hope I did not alter your name too much.
Not yet.
It's yours.
Well, thank you so much, and thank you so much, Jenny, for the invitation. I really appreciate that. I'm very impressed with Biogem and all the people who have been working and are working at Biogem. And we are really looking forward to work with you and collaborate with you in a variety of different projects that I am doing, as well as other investigators who are giving a talk today. So to get started, let me see if I can get it started. Okay. So my lab really is divided into two different groups of people. Part of people in the lab, they are working on non-coding RNAs and specifically microRNA and long non-coding RNA. And the other part of the lab, they are working to define the role of mitochondrial structure in the progression of diabetic kidney disease. And at the end of the day, based on either mitochondrial dynamics, signal transduction, or the role of RNAs, we are really trying to converge all those data into figuring out what is the role of mitochondria on the kidney homeostasis, and also when you have diseases such as kidney disease and diabetic kidney disease. So today I'm going to talk briefly about mitochondrial dysfunction in diabetic kidney disease, just as a background, and then something that I'm hoping that you would be interested to help my lab in terms of targeting mitochondrial fission and the important protein which is critical for mitochondrial fragmentation, which is DRP1. I'm going to talk briefly about a microRNA 93, and then something that we have been working for the last 5-6 years, which is the role of a long non-coding RNA, another non-coding RNA, as a signature non-coding RNA in diabetic kidney disease. So what is mitochondrial dysfunction? I think most of us agree that mitochondria really works like an engine for a car, and if the engine is not working, obviously the car would not be able to function properly. And as a result, mitochondrial dysfunction has been shown and recognized to play an important role in the development of diabetic kidney disease, which is the key disease that we have been focused on for the last 25 years. And although we know that mitochondria and mitochondrial dysfunction is important, we really don't know what's the nature, what's the definition of mitochondrial dysfunction in diabetic kidney disease, and what's the value of targeting mitochondrial dysfunction in terms of mitigating progression of diabetic kidney disease. So today I'm going to talk a little bit about our last work that just got published in Nature Communication based on what happens if we target mitochondrial electron transport chain. What happens if we really directly target the engine of the car to see whether we can replace the engine and what kind of mileage we get out of that if we can replace the engine of the cell. In other words, for us, kidney diseases is kind of, we have a very mitochondria-centric view of at least diabetic kidney disease, but most likely in many other types of kidney disease. And what we define diabetic kidney disease is really a journey, is really a cascade of dysfunction from an organelle, which is mitochondria, to an organ, which is kidney. So I'm going to take you to a journey of how dysfunction of mitochondria, dysfunction of organelle, could lead at the end of the day to an organ dysfunction and kidney dysfunction. So when I joined MD Anderson, we were thinking that, well, maybe cancer, diabetic kidney disease, and the process of aging, they really don't have much in common. But we soon realized that mitochondrial dysfunction has been recognized to play important roles in all of those three processes. So in terms of cancer, I think some of you guys who work in oncology definitely are familiar with the concept of Warburg effect. And the concept is, very simply put, is that the glucose gets into the cell and undergoes glycolysis. If you have oxygen, you get pyruvate, and from pyruvate, then you go into TCA cycle and you generate ATP. So basically, you extract ATP, which is the chemical energy of the cell, from nutrients through in the mitochondria. But in cancer cell, rather than using this pathway, which is pathway of generating ATP in the mitochondria, they use the other pathway, which is the pathway for glycolysis, and they develop lactate. So Dr. Warburg came up with the idea that, well, there must be a problem with mitochondria, because why would it be that tumor cells, rather than using mitochondria and generating ATP, they target glycolysis and they go through glycolytic pathway? There must be a problem with mitochondria. And if you can reverse the process in mitochondria, probably you would be able to cure cancer. I think more importantly to the talk that I'm giving today, Brownlee and his colleagues in the early 2000s, they came up with the idea that there is also a mitochondrial dysfunction in all the complications of diabetes. And their idea was based on the notion that at the end of the day, there is increased mitochondrial reactive oxygen species. And even though there are multiple pathways that are known to play roles in the development of diabetic kidney disease, the main source of all these pathways is mitochondrial ROS. And if you can reverse mitochondrial ROS, you probably would be able to reverse progression of diabetic kidney disease. So we started to work on the definition of mitochondrial dysfunction in diabetic kidney disease about maybe 20, 25 years ago. And the idea that we put forward initially was, what is the role of mitochondria in the first place? What do they do? And when you really think about, in addition to the main function, which is the bioenergetic effect, which means that they generate ATP, what else mitochondria are doing that could be important for a disease process? And when you think about it, there are many other types of functions that mitochondria are able to do. One is that, at least for 50, 60 years, is known is the effect on biosynthetic effect of mitochondria, which means that they generate metabolites. And those metabolites, usually they are in the TCA cycle, Krebs cycle, which is also tie mitochondria with signaling pathway, because once mitochondria generate some of these metabolites get out of the mitochondria, they go into the cell, and they have a diverse functions in the cell. So it's really mitochondria, when you think about it, it seems that they work in a way that seems to be a signaling molecule. It's a hub for different signals through metabolites. But also they're important for reactive oxygen species, for calcium, obviously apoptosis. Many people in the beginning, when the idea that mitochondria is important for apoptosis, they didn't believe it, because they say, well, why would it be? Why would that mitochondria would be important for apoptosis? But nowadays, we accept it as a fact that this is a part of the function of mitochondria. So in about 10 years ago, we came up with the idea that one of the features of mitochondria in diabetic kidney disease is that when you look at podocytes, which are like a specific cells that Dr. Fornoni just mentioned, you look at mitochondria, they seem to be fragmented. And here you can see basically normal mitochondria, and these are mitochondria in podocyte in a diabetic mouse model. So then the idea is that, well, why is that? Why is that when you look at mitochondria in the kidney, a podocyte, they are fragmented? Now, I remind you that mitochondrial fragmentation, it's something normal, happens physiologically all the time. And when the cell tries to divide, mitochondria, obviously, they have to divide as well. So there's nothing special about fragmentation of mitochondria. But in diabetes, there is an increase, there is enhanced mitochondrial fission. And that's very important, because once the cell undergoes an excess of mitochondrial fission, or fragmentation, then they're going to have more mitochondrial ROS. This is kind of signaling to the cell that these mitochondria are not working well. And then cell tries to kill those mitochondria. And the way it happens is through this molecule that's called DRP1. So DRP1 is basically a molecule, a protein, that is just minding its own business in the cytoplasm. And when it gets this signal from the cell that mitochondria should undergo division, goes around mitochondria and strangulates mitochondria and kind of helps to divide mitochondria into two. And what we found was that in diabetes condition, and here we call that hyperglycemia, in contrast to the normal condition, there are some kinases that are important for phosphorylation of this protein, DRP1. And once DRP1 gets phosphorylated, goes around mitochondria, strangulates mitochondria, and fragments, or the mitochondria really undergo mitochondrial fission. So that was the initial proposal that we put together, that somehow in the diabetic milieu, in the diabetic environment, you have an excess of mitochondrial fragmentation. That excess of mitochondrial fragmentation at the end caused increased mitochondrial ROS and ultimately leads to killing of the cell, and that's why you have less podocytes in diabetic kidney disease, because the cell undergo apoptosis. So then we tried to test this hypothesis, and we said, well, if DRP1 is so important, if we have a diabetic mouse, and presumably this diabetic mouse has increased activity of DRP1, what happens if you knock out DRP1? So diabetic mouse doesn't have DRP1 anymore. So idea was that if that happens, you probably should see some improvement in diabetic kidney disease. Now, when we knock out DRP1 in non-diabetic conditions, we saw that actually nothing happens, which is kind of surprising, because if you knock out DRP1 embryonically, the animals die. But if you do it later on, nothing really happens to the mice, which shows that there are other mechanisms that they do the job of DRP1 in terms of mitochondria. But surprisingly, if you knock down or knock out DRP1 only in podocytes, and you make the animal diabetic, and here we made DB mice, this is an established model of type 2 diabetes, and you look at, well, what happens to albuminuria in these mice, you see that if you knock out DRP1 in podocyte of this diabetic mice, you see improvement in the amount of albuminuria, which really means that DRP1 phosphorylation is important for progression of diabetic kidney disease. We also look at the mitochondria in these mice, and mitochondria, they look completely normal in contrast to diabetic mice. And the last thing we did was that, well, we said if DRP1 gets phosphorylated, and this phosphorylation of DRP1 is important for its activation, what happens if we knock out that phosphorylation site in a mouse model? And that's what we did. We mutated this serine phosphorylation to alanine. As you can see here, we made diabetic mice. If the mice are not diabetic, the same thing, nothing happens to the mice. But once you make animal diabetic, and then you look at what happens to amount of albumin in the urine, as you can see here, you also see significant improvement. This is a diabetic mouse, or there are 5 diabetic mice here. But when you make a mutation in the serine phosphorylation, you don't have that much of albuminuria anymore, really indicating that that phosphorylation site is important. Now, what we have done afterwards was that, well, what is the best way that we can control what's happening to mitochondria? And we thought that, well, the main function of mitochondria is obviously generate ATP. And ATP comes from electron transport chain. If you think about biochemistry 101, I remind you that basically we have 4 different types of mitochondrial complexes that they transfer electrons. And each time hydrogen gets released, at the end, you have ATP synthase that generates ATP. So we wonder, like, well, what type of changes you see in mitochondrial complexes? What kind of problems you see in the engine of the car when you have a diabetic mouse? And when we did that, we did a proteomic assay to look at all the proteins in all these complexes, what happens to them in an animal model. And when we did that in complex 1, which is the entrance part for electrons to get into electron transport chain, in diabetic mouse was significantly lower than control mouse. So somehow complex 1 is not working, it's kind of like working in a lazy manner as compared to other complexes. And then we really focus on a subunit of complex 1, which is called end of S4. And just we chose that because we saw that end of S4 is much lower, not only in diabetic mice, but also in patients with type 2 diabetes. And we said, well, maybe there is something special, magic about end of S4 that is consistently lower as part of complex 1 in all types of diabetic environments. So here, I'm just trying to show here, when you look at the different stages of diabetic kidney disease, you see independent of the stage, you have significant reduction of end of S4. So it's not like we are just trying to cure diabetes in animals, in mouse models of diabetes, but maybe there is something special about end of S4 in patients with diabetic kidney disease. And most of the time, since we work on animal models and we generate animal models, we said, well, if end of S4 is low, there are just 2 reasons for low end of S4. One is that because you have an excess of glucose coming to the cell, as an adaptive mechanism, complex 1 gets kind of like deactivated in a way to protect the cell. Or complex 1 is deactivated just because it's pathogenic. So low levels of complex 1 could be something pathogenic for the kidney. So in that way, if you overexpress something with complex 1, you should see improvement in diabetic kidney disease. And we overexpress end of S4 in podocytes of animal models of diabetes. And once we did that, and we looked at different things like albumin in the urine, you can see that these are basically control mice, and this is a diabetic mice. And if you make this transgenic, you see significant improvement of the amount of albumin in the urine, which was surprising to us, because why is that you just overexpress just one subunit of a complex, and complex 1 has 46 different subunits, but we just chose one of them by chance. We overexpress the subunit, and we see changes in the phenotype in a very complex disease. It's sometimes things are just too good to be true. So we looked at it, well, what is the reason that this subunit, when you overexpress that, you see improvement in the phenotype, and whether this is even true. So we looked at mitochondria first, and you can see that diabetic mice, as expected, they had this fragmented mitochondria. But when we overexpress end of S4 mitochondria, they looked much better. So somehow you just overexpress subunits of complex 1, And it's kind of like you are repairing the engine of the mitochondria of the cell or of the car, and somehow the car now runs much better. Now, the idea was that what is the mechanism? So I think we have shown several times that end of S4 is low. You overexpress that, you see improvement in diabetic kidney disease. You see improvement in mitochondria. But why is that? What is special about end of S4 that make this happen? And as we were looking at different parts of mitochondria, we realized that in diabetes, when you look at diabetic mitochondria, not only they are fragmented, but also when you look at cristae, which is like this imagination of mitochondria, they are completely distorted. Whereas with end of S4, you see that not only mitochondrial structure look much better, but also this cristae formation, they look normalized. And that we thought, well, maybe that makes sense, because if you improve cristae, then you can stabilize mitochondrial complexes that they sit on cristae. And if mitochondria, they're sitting where they're supposed to function, then maybe electron transport chain is one functioning better. And to test this hypothesis, we did something called proximity labeling, which really looking at what are the proteins close to end of S4. How does end of S4 or where end of S4 is binding to proteins? And once we did that, not surprisingly, we saw a lot of proteins that were part of the cristae formation. So we saw that the end of S4 or is excess of end of S4 somehow binds to some of those proteins that are important for cristae formation. And to make a long history short, and we looked at this phenomenon maybe a little bit in a fancy way, but using very fancy electron microscopy as well as storm and STED microscopy. But at the end of the day, we focus on a protein called STOML2, that it seems that this protein, which is part of the cristae protein, binds to end of S4, and once they bind to each other, they kind of stabilize the cristae formation, and then the mitochondrial complexes, they sit on it, and then what happens is that electrons very nicely pass through these mitochondrial complexes, hydrogen gets excreted, and you have more ATP. Now, because I have published part of this work, I'm not going to spend too much time on it, and that was published not too long ago. So this is the conclusions that we had. When you look at the cristae formation, in normal condition, as you can see here, or basically in diabetic condition, let me say here, you have less end of S4, you have less interaction between end of S4 and STOML2, and you have mitochondrial remodeling because cristae get destabilized, and you have podocyte injury. But when you overexpress it, you can reverse many of these features. So one of the things that we are thinking, and probably we need your help for that, there are two things, because what would you do next? Now you have a description, you have something that seems to be important, you have a protein that seems to really make a difference in terms of the phenotype, but what would you do next? And we have done two things, and I'm looking forward to some of your suggestions. The first thing that we did, we made a drug discovery, and this drug discovery end of S4 reporter, the way it works. So if you have a, let's say, hundreds, thousands, millions of small molecules that you add to this construct, because this is based on a GFP assay, you can see what kind of chemicals can increase or decrease end of S4. And then you can use that idea and test the hypothesis whether that is the case or not. And the second thing we did, we thought, well, maybe targeting end of S4 is complicated. So we did something that is called CRISPR-I or CRISPR-A, which is a genome-wide approach, either inhibiting or activating end of S4, just to see what other genes, in terms of, in a genome-wide fashion, can modulate the effect of end of S4. And once you have that, not only you can just target end of S4, you can target anything else that has any effect on end of S4, either decreasing it or increasing that. So let me just say a few words before I think just a few more minutes. Just microRNA, because some of you guys I know working on epigenetics. So microRNA, we worked about maybe 15 years ago. I think all of you guys know that microRNA is a single-stranded RNA, about 20 nucleotides. We have extensively worked on the role of microRNA. Our initial work was that there is a microRNA 93 that seems to be downregulated in diabetic kidney disease. And as we usually do, we overexpress miR93. And we saw that when you overexpress miR93 in diabetic mice, and you look at the amount of albuminuria in these mice, you see that basically there is significant improvement in the amount of albuminuria in these mice. And here you can see that basically that if you don't have diabetic mice, you don't have any changes. But if you make animal diabetic and you overexpress miR93, you see a significant change in the amount of albuminuria. So somehow miR93 is low in diabetic mice, you overexpress it in podocyte, and you see improvement in diabetic kidney disease. We really have not worked on it for the last 7-8 years, because we got to something that we thought was much more interesting, and that's the role of long non-coding RNA or link RNAs that by definition are more than 200 nucleotides. I'm not going to go over the definition of link RNAs, except that they're very similar to mRNAs. As you can see, they are capped, they have poly-A, but they are not exactly like mRNA because they are cell and tissue specific. The expression level is much lower. And also, by definition, they are not supposed to have protein coding region, even though that has been challenged more recently. So we looked at all the link RNAs that, as compared in diabetic glomeruli, as compared to wild type, and what we found was that one of them, called TUG1 or TOG1, is significantly reduced in diabetic mice. And this TUG1 is mainly nuclear, not so much cytoplasmic. And when you look at the diabetic mice, you can see that almost of this non-coding RNA. We overexpress this long non-coding RNA. And as you can see, when you look at it, this is just proof of principle that we overexpress this non-coding RNA. And when we looked at the amount of protein in the urine, and that's the way we looked at the phenotype of mice, you overexpress TUG1 in the podocytes of diabetic mice, and just by doing that, by overexpressing a link RNA, you see improvement in the phenotype of diabetic kidney disease. And like always, when we do this kind of work, you always ask, well, how does that work? You overexpressing a non-coding RNA, you see improvement in diabetic kidney disease, but what is the mechanism? And the mechanism that we got interested, we found that is through a protein called PGC1 alpha, which is a regulator of mitochondrial function and biogenesis. And if that's the case, when you look at mitochondrial biogenesis, or mitochondrial copy number in diabetic mice. If you overexpress TUJ1, you can see that there is significant increase in mitochondrial copy number. So that would be consistent with the effect of TUJ1 through PGC1 alpha. And then we did something that's called ChIP-seq analysis. We looked at genome-wide binding site for TUJ1 to see, well, where does TUJ1 is binding to DNA? And what we find at the end of the day was that if this is TUJ1, on one hand binds to this part which is called TUJ1 binding region, and on the other hand it binds to the PGC1 alpha itself and brings down PGC1 alpha to its promoter region. And by doing that, it accelerates transcription of PGC1 alpha. And at the end, it's kind of a little bit complicated, but the question we had at the end was that, well, we know that PGC1 alpha seems to be downstream of TUJ1, but how can you prove that? Well, the only way to prove that is that if you have a diabetic mouse, which has a lot of protein in the urine, you overexpress TUJ1, you should see improvement in the amount of albuminuria. And then if you knock down PGC1 alpha in the same mouse, you should lose the effect of TUJ1 on albuminuria. It seems very, very complicated, and it is complicated. It took us almost 2 years to generate mice that there are 4 different mutations in the same mouse. But by doing this, when we, at the end, after 2 years we did this, and we looked at our mice, you can see in the diabetic mouse, as you can see, there is increased amount of albumin in the urine. If you overexpress TUJ1, you see improvement. But if you, in the same mouse, if you overexpress TUJ1, but you knock down PGC1 alpha, you lose the protective effect of TUJ1. And also we found that the way it works is through a metabolites in the mitochondria in a way that PGC1 alpha goes up, arginine goes down, ornithine and citrulline goes down, and that happens to mitochondria biogenesis that goes up, fission goes down, and ROS goes down. And when you have the PGC1 alpha knockout, you see the reverse process. So I'm going to stop here, but I think in this regard, what we're really interested is mostly not so much in terms of how TUJ1 working, but is there any way that we could have some sort of a clinical trial that we could use substrate that we know they increase the amount of TUJ1 based on our work in the last few years, and see, well, what happens to patients with diabetic nephropathy if we give them substrate to increase the amount of TUJ1? Would that really make a difference in those patients, or is it just we are treating diabetic mice rather than our own patients who have diabetic kidney disease? And with that, I'm going to stop. I'm looking forward to your question or suggestions. Thank you.
So, questions for the audience? Vincenzo?
Thanks a lot for your presentation. So, if I understood well, mitochondria and their dysfunction has an impact on disease. So, I was wondering if it would be useful to study the possible involvement of regulated cell death pathway, not apoptosis, in this dysfunction, mitochondrial dysfunction, and so in pathogenesis. And maybe it could be a possible target for the therapy.
What do you have in mind?
Okay, so I am not an expert of kidney and diabetes, but I know that several of them are involved. For example, necroptosis, for example, paroptosis, and in particular, last year, raw evidence are about necrosis.
Yes.
Nets, so neutrophil death, have an impact on kidney disease and maybe also in diabetic disease.
I think that's an excellent suggestion. I think in reality, the way I at least take a look at it is that all of those processes, somehow they are interrelated. So I wouldn't be surprised that you see changes in all of them. But I think in this case, we should have maybe a holistic view of what are we trying to show. It's not maybe so much the signaling pathway, which is important, unless you really want to target a signaling pathway causing apoptosis or any of those, but rather maybe something upstream of that. So you can maybe modulate all of those pathways at the same time because you're targeting something upstream of all these smaller pathways that then you should see. I mean, that's almost a given that you should see some of those changes in the podocyte of these mice. Yes.
Yeah.
When many studies have results about MYO93.
Yes.
And when you talk about the chance of a replacing strategy with nanovectors or nanocarriers delivering the MYO93 in the podocytes of the diabetic animals and then also
Yeah, that's also is a very good question. The reason we kind of quit that idea of targeting MYO93 was basically twofold. One was that there was a paper that showed if you overexpress MYO93, that may cause cancer. Now, you have to be careful when you overexpress microRNAs because obviously if you overexpress it 100-fold, who knows what will happen to those microRNAs. Yeah, yeah. Yeah, yeah, I think definitely that's the case. Yes, I think definitely that is the case, and we could have done that. I think that's something. Most likely, I think if we do that, we shouldn't really see any signs of cancer if we deliver it to podocyte or at least kidney. That should not be the case, especially if we don't increase the amount of MYO93 like 100-fold more than what it's supposed to be. If we increase that 10-fold even, I think that should be good enough to see some phenotypic changes. But the other reason was that when we were working, and I had a graduate student who was working on MYO93, another graduate student who worked on this link RNA, and his results were spectacular, quite frankly. And we had some substrate that we can use as drugs to really increase the amount of that link RNA. And we thought that maybe would be a better way of looking at the role of non-coding RNA rather than MYO93. Having said that, I think it's not a bad idea that we revisit this idea of the role of MYO93 because I know some other people, they have looked at the effect of MYO93 and VEGF is a downstream effect of MYO93 and they have used it in diabetic retinopathy and they have had great results with that. So maybe that could be the same thing for diabetic nephropathy. We haven't done it. But if somebody is interested, they are more than welcome to, we would do all the constructs we have. They are more than welcome to all of those constructs.
The second question is about methodology. Do you think that the application of methodology in the diabetic podocyte can control the disease?
Yeah, I think definitely is the same thing because we are looking at just a small puzzle of the disease and somebody else is looking maybe on mitophagy, but all of those things are interrelated. So it's not like we get just like one problem is going to solve it and there is no mitophagy. We are looking more something upstream that if you reverse that process, you most likely can change mitophagy as well as apoptosis, as well as other things that are happening at the same time in podocytes of diabetic mice. and you can reverse the phenotype of the mouse.
Last question is on single cell RNA.
Single cell RNA? Oh, definitely we would be interested. Nowadays, if you don't have single cell, you cannot publish in any reputable journal, just the way it is these days. So that's something that you do. You're saying that that would be a better way of looking at this. I think definitely that maybe we can talk after this presentation, because that would be an interesting. We can do single cell ourselves, but I think that it's almost impossible these days to do all the techniques yourself. And there is no point to do that really. So it's better like different people do different ways of, and then we get together as a team and really use the result of single cell RNA, as well as the other things that we are doing. Yeah, it would be fantastic if we can work together, for sure. Yes.
Let's put together two main presentations. The first observation is yours. You say that in the pathogenesis of diabetic kidney disease, the kidney solve, there must be an involvement of mitochondria. That's it. And you have to solve the diabetic, even if you have some possible targets. On the other side, we know that actually, in clinical point of view, we have a lot of new agents that cure diabetic kidney disease.
Yes.
But we don't know why.
Yes.
Any thought that the mitochondria could be involved in the effect of these antigen that SGP2 that factor they reduce the sequence of the kidney involvement?
I have no doubt that all of those agents, they do have an effect on mitochondria. Whether the effect is a key effect or happens as an innocent bystander, that I don't know for sure. But there is no question that they have some effect. The least would be maybe in terms of like energy, bioenergetic effect of mitochondria, you see some improvement. But some improvement, does it mean, is it good enough? Or the cell needs even more ATP to really get out of the state that they have been already there. But all of those things could be tested. These are all testable questions that could be done. It's not that difficult to see what's the effect of SGL2 inhibitor, I'm just making it up, on mitochondria. And that would be a very easy and testable question that we can do. Would it make that much of difference? You can argue no, because SGL2, you already know that it's going to work. But it's through mitochondria, ER, okay, good, but I mean, does it make that much of difference? You can argue yes, because if you can find a target in mitochondria that does play a major role, rather than using something that is not that precise, it's not that you can use something very precise for what happens to progression of diabetic kidney disease. In that regard, probably that would be contribution to what we know about diabetic kidney disease. So I'm not saying don't we do it. It's hard for a lab just to go ahead and just try everything related to mitochondria and diabetic kidney disease. But that's why we have so many young investigators that would be, these are easy experiments to do. These are testable experiments. And within few months, at least at the cell level, they can have some result, and then we can validate some of those results in animal models and then in patients even.
I also have a small question. So you showed convincingly the story of LAF4 and the fact that if you overexpress it, you somehow have at least a kind of phenotypic amelioration,
Changes,
something like this. So first of all, is why is it downregulated in the model or in the patients? So is it something related to hyperglycemia? And if you would have followed an opposite strategy, so in normal, let's say, models, you downregulate LAF4, would you have a phenotypic deregulation in the kidney, in the podocytes, or in the models?
Well, these are extremely good questions. So there are some patients that they don't have any end of S4, and they die at the age of 3-4, because this is such an important protein for mitochondrial function. But we are not talking about completely knocking out end of S4, obviously. We're just reducing the amount and the activity. Is there something special about end of S4 that the other subunits they don't have? My guess would be that's not the case. I think if you look at some other subunits, and if you overexpress it, if they are downregulated, probably you should see some improvement in kidney function as well as the structure of mitochondria. Probably that doesn't happen with all the subunits, but some of the subunits that play important roles, I think if you play with those, probably you do see some improvements, for sure.
So why is it downregulated?
Yes.
Something the promoter?
I mean, the idea that we have, and that's a very, we thought about it very carefully. In the beginning, we thought that, well, just think about a factory that you put a lot of substrate into the factory. So mitochondria, they don't need so much of glucose. If you put a lot of glucose into the cell, and mitochondria are smart, they try to downregulate everything because they don't need that much of ATP. So what they do, you have a lot of substrate coming to the cell, but these mitochondrial complexes, they work very slowly. So that kind of like protects the cell from high glucose environments, right? So if that was the case, if that would have been the case, if you express these complexes, you should see worsening of kidney function, because these are like adaptive mechanisms. Our results show that's not the case. So these problems are primary problems. It's not just because you have so much of high glucose that this happens. But your question is still valid. Well, why are they low anyways? And the one reason and one way to answer this question is look at, well, what happens to genes that can regulate the amount of end of S4, either in high glucose and normal glucose condition. A genome-wide screening of all the genes, potentially all the genes in the body that can affect or modulate the amount of end of S4. And to do that, we do have to do CRISPR-I and CRISPR-A, because that gives us some clues, like, well, at least some pathways. Well, what's going on? So why end of S4 or other complexes are so down and so down-regulated? Even if it's because of hyperglycemia, there must be through some links. It's not like hyperglycemia directly does that.
But it could be maybe half-life of the RNA or a half-life of the protein, modification of both, for example, could be a way.
That definitely would be a way, but we saw that the effect of glucose is selective. It's not that all the subunits of complex 1 are downregulated or the subunits of complex 2.
More like on a, let's say, scale down. So some
Stepwise.
Stepwise. Not the glucose or the hyperglycemia, but maybe some complexes at the mitochondrial level that could maybe act, since you don't need to act on the half-life or something like this. There are these mechanisms, maybe adaptive or not, or maybe you are also acting on some additional targets of mitochondria, such as, I don't know,
I think definitely
methylations.
I couldn't agree more with you. And actually in our discussion, we put some of those ideas. But in journals nowadays, they say, well, you don't have any data to show that this idea are correct or not. You're just saying things that without showing any data. But I think it's important to extrapolate sometimes your data and put some of those ideas that we think that could be, and maybe we are wrong, and there is nothing wrong with that. But, it's not like whatever hypothesis we have should be correct.
So in hungry cells, so starved cells, maybe even podocytes.
Yes.
How is this situation? So let's say if they are hungry, or let's say with a low glucose level, so how is this situation?
I think at the end of the day, mitochondria, they respond more or less the same way to stress. Either too much glucose or not enough glucose. The cell at the end of the day, they die.
The same stress.
yeah, mechanistically may be different, but at the end, what you see as a phenotype probably would be the same. And as the other gentleman said, all these processes, they are interconnected. So it's not like we can just focus on one and forget everything else, because body is very complicated, especially when it comes to complicated diseases such as diabetic kidney disease. And with this, I see that Raphael is getting a little bit upset with me. I'm just gonna.
No other questions? Okay, thank you.
Thank you. Thanks.
Very nice discussion. Very nice discussion. I just want to make a comment. I can see that the program of Saturday on DKD was really very well assembled, because we spoke about mitosis, we spoke about mitophagy, and finally everything come up again with your presentation. But now it's a pleasure to me to present you Professor Raphael Kramer, that is a professor of medicine at Aachen University in Germany. His lab is really one of the leading labs in the field of fibrosis, both the kidney and heart fibrosis. His lab is expanding and changing actually the perspective in the CKD. We were all mainly focusing on what is before fibrosis. Now we are with his lab, we are learning more and more of how to control fibrosis. And also, his lab and other labs in the world are now changing the paradigm of using the right sample. We are going from experimental model sample to human sample directly. And kidney biopsies is not becoming any more useful for diagnosis, but also for research. And I'm sure that in the next future, we can expand the possibility to make diagnosis on kidney biopsy by using one of the techniques that I'm sure Raphael will speak about. So I don't want to take more minutes.
Thanks so much. So do I have to share my screen for the Zoom, or is this all set? It works? Okay. Okay, so I think it's enough about data extrapolation from Texas. So now we're going to talk about some other stuff. Yeah. No, so I'm really glad to be here and really enjoyed, the entire day. We had fantastic food, but also I enjoyed really to see your labs. I think you have really set up something amazing here. And I think there can be a lot of, opportunities to collaborate. So my lab is really focusing a lot on fibrosis. So just my disclosures. And on kidney disease. So just as a nephrologist, and you have some nephrologists here, I always like to, highlight this. And when you look at the global mortality causes, by 2040, chronic kidney disease is estimated to be number 5 killer in the world. In Europe, the prevalence of CKD is 10%. This is pretty much all over the world. 1 in 3 adults are at risk. When you look at this, CKD globally accounts for more years lived with disability than all neoplasms combined. So then you really realize that this is an important disease, that we have to do something there to really tackle the disease. And when you look at nephrology, we're really lagging behind, or the kidney field is really lagging behind in the number of randomized controlled clinical trials. It's really here at the very bottom compared to most other organs. And that's why we still have very limited treatment options. I mean, we have now some new drugs, SGLT2 inhibitors, etc., that work, but still the majority of patients, even under clinical trial conditions, progress with their disease. So my lab is really studying fibrosis, and fibrosis is basically scar formation. It's very similar. When you cut into your skin, you get a scar, right? And this happens in all organs. This is the kidney. All this blue stuff here is scar formation, and these fibrotic fibers basically destroy the architecture of the organ and then lead to organ failure. And this is also the same in the heart. This blue stuff here stiffens the left ventricle. You get stiffening. The heart doesn't pump as much. And it also causes electric instability, so sudden cardiac death from arrhythmia. It has been very controversial. Where do the cells come from that drive fibrosis? Whether these are resident
fibroblasts or parasites, whether this is epithelial cells, endothelial cells, circulating bone marrow-derived cells such as fibrocytes. And we have done some work, and this was really driven by Christoph and Mahmoud from the lab. They both transitioned now into independent positions to use single-cell sequencing of human nephrectomies to understand which cells become myofibroblasts. And you can see in this model, and this is a diffusion map, and we did a lot of models and validation experiments to really validate that there's parasites and two distinct fibroblast lineages that differentiate to these myofibroblasts. So when you look at ECM scores, so this is just a gene set for all extracellular matrix genes, basically. So proteoglycans, glycoproteins, and collagens, you can see it's really not highly expressed when this starts this diffusion map, and then it's very high here. Same, you can see this even better for collagen 1. So these are the major contributing cells. But then, you know, this is single cell sequencing, and I like to, you've probably seen these little images here very often. I like to use it to show you what's the difference between single cell, bulk transcriptomics, and spatial. So this is basically how we do, or how we have done this a lot in the past. So microarrays or bulk transcriptomics, it's really like a smoothie. So you don't know which gene comes from which cell. You just see have a soup of genes that are higher or lower. You don't know how much banana, how much strawberry is in there, but you have single cell sequencing, and you guys are doing this here too. You can really see how many pineapple pieces, how many strawberries are in there. You can look at the strawberry and check it out, but you still don't know anything about the spatial relation. So spatial transcriptomics is really more like a fruitcake. So you know exactly what's the spatial relation of the pineapple to the strawberry. And so we have done this work early on where spatial transcriptomics, this Visium pipeline from 10x Genomics wasn't commercially available. We had a collaboration with the company, and we thought, okay, which kind of disease would be interesting to use to explore really the capability of the spatial transcriptomics, and that's why we actually went for human myocardial infarction. My lab really studies also the heart. We thought, okay, when you open the textbook in medical school, what disease has really like a major spatial component? It's the heart, and the resolution of this technology wasn't there yet. It's getting there. It's really subcellular now, but wasn't there yet to really study the kidney. And the kidney, in a given spot of this technology, and I'll show this on the next slide, you would have 10 to 20 cells, and in the heart, it's just 3 to 5. So, so we had actually human hearts, and this is from a total artificial heart program in Bad Oeynhausen. It's the largest German heart transplant center. They had actually patients that came to the hospital in a cardiogenic shock. They actually put in like a total artificial heart and then explanted the heart for research. And so we have 4 donor. These are non-transplanted donor hearts. This is as healthy as you can get human heart. I couldn't convince any of my postdocs or students to donate some heart tissue. So, and then we have different time points after injury. So this is early after myocardial infarction. There you can look, you have ischemic zone, there's really cell death. We have a border zone here, there is some injury, there's some surviving cardiomyocyte, and the remote zone is not injured. And then late after myocardial infarction, you have fibrotic zone, you have remodeling, fibrosis, scar formation. And so what we did is actually we took sections, 10 micrometer slices for this Visium slice. So these Visium slice, you have 6.5 times 6.5 millimeters. And on these capture areas, you have 5,000 spots, and every spot has a diameter of 55 micrometer. So now there's like better technologies. So sequencing more. It's a bunch of new technologies, fish-based, in situ sequencing based. There's nanoball sequencing technology where you can now go to subcellular resolution. But this was the technology then. So 10 micrometer slices for spatial, and then the adjacent tissue, we sorted nuclei. So this is just DAPI to isolate nuclei, and the nuclei we used then for single nuclear RNA sequencing and single nuclear ATAC sequencing. And the idea was, because we don't have the cellular resolution here, we can use these 2 layers to integrate the data and then computationally increase resolution. And so I'm just going to walk you through some slides. And when we had them early on, we were quite excited about these. Just some spatial clustering here, non-supervised clustering. You see there's a lot of different zones here, and you can see the hematoxylin eosine staining here from the acute infarcted human heart. it's really nicely what you can see that there's still some intact tissue here. Then there's a lot of holes in the tissue, so there's a lot of cell death in these areas, a lot of eosine positive cells here. There's a lot of immune cells coming in, so you can see the myocardium is quite intact here, but really destroyed in this area. So now when you look at gene counts, you can detect in these areas where you still have healthy myocardium, you can detect up to 10,000 genes in the red areas, but not that many in these blue areas. It's just 500 to 1,000 genes because there's so much cell death. So now just some marker genes, very boring, but troponin, you know this from the clinic, it's a biomarker for myocardial infarction. So there's some surviving cardiomyocytes, specific for cardiomyocytes that are here. There's less surviving cardiomyocytes in this area because you have cell death. FTH1, a macrophage marker gene, and this is now really like when you look in your textbook, there's demarcation of immune cells of this acute infarcted area. These immune cells really demark, they come in, they eat the dead cells and the debris, really like when you look in the textbook. And then there is collagen 1 alpha 1. This is a fibroblast marker gene. Obviously, there's early myofiber or fibroblast activation, which is obviously very important after myocardial infarction because you need to close the wound. Otherwise, you would have left ventricular rupture. CXCL8, this is a granular site marker gene. So many of these nuclei you could see in the hematoxylin eosine stained image here on the bottom left or bottom right from you. These are granulocytes coming in. But so now what can you do with all of these three layers? So spatial transcriptomic, single cell RNA, single cell ATAC. So you can really look at entire pathways. And this is just an example for the TGF-beta pathway. So you can actually map this pathway to space. And what you can see here is actually a U-shaped scar 100 days after myocardial infarction. You can see within the scar there's tremendous activity of the TGF-beta signaling pathways. This is now using a tool called Progeny. You can estimate pathway activity based on perturbed genes. So when you perturb a certain pathway in a cell line and then measure which genes are downstream expressed of the pathway being active, and there's a lot of open source data in this relation, you can estimate based on gene expression which pathway is active. You probably know these some other tools like CAG or PID, where you can estimate pathway activity based on members of the pathway that are being expressed, but it's very unprecise because being a member of the pathway and being expressed as a member doesn't mean the pathway is active. So you can see there's really increased TGF-beta signaling, and now you can look at the entire pathway. So you can look at the transcription factors of this pathway. And this is now actually using the ATAC sequencing data. So the ATAC sequencing data allows you to estimate transcription factor activity much more precise compared to mRNA data because you do footprinting. So you basically know which DNA binding motifs are needed for certain transcription factors. And you can look at the accessibility, so at the open chromatin of these DNA binding motifs, and then you can actually map this to space. So this is actually really precisely in space now estimating activity of this transcription factor, which is downstream of TGF-beta. And then you can look at genes that are regulated by this transcription factor and also map them to space. So what you can also do, you can increase the resolution computationally. So what we did is actually with the single cell sequencing data, we ask in a given spot, and these are all the different spots, which genes in this given spot, and I mentioned this is 55 micrometers in diameter, so there's several genes in a given spot or several cells in a given spot. We could estimate which genes in this spot are coming from which cells. And what you can see in the scar here, there's most genes here coming from these fibroblast subset here that really drives scar formation. And around the scar, you can see that here there's most genes coming from this endothelial cell 1 population. So these endothelial cell 1s really, they drive neoangiogenesis around the scar, which makes a lot of sense. So now you have all these spatial data sets from all these patients, and how can you analyze this to really gain biological useful information, and so to understand mechanisms, identify novel drug targets, etc. So what we developed is actually with some collaborators, some machine learning algorithms to analyze these slides. And what this data showed us, and this is just one example, is like that the presence of fibroblast in space always predicted the presence of immune cells and vice versa. And also, this is just another example, the presence of fibroblasts in space predicts the presence of TGF-beta signaling activity. You can do this with all pathways, all cell types, etc. Really sieve through this and try to understand what is happening. I'll show you an example of this later. So as I mentioned, my lab is really interested in fibrosis. So we really look at heterogeneity of fibroblasts in the human heart. There's 4 major populations. We always see these SCARA5 positive non-activated fibroblasts. These are present in all tissues. They're present in the lung. They're present in the liver. They're present in the kidney. These are non-activated fibroblasts. And then we see these periostine positive myofibroblasts that are always present in fibrosis, really across organs. So we developed this computational model, very similar to what I showed you in the kidney. This shows us the pseudotime differentiation of these SCARA5 non-activated fibroblasts towards these myofibroblasts. They're called fibroblast 2 here. So ECM score, I introduced this, all genes that are associated with extracellular matrix, low here, high here. And then you can look actually at space. You can see SCARA5 cells are around this scar. They are inactive, and once they get activated, they migrate into the scar, express periostine, fibronectin, collagen 1. So now this is just a computational model, and so how are you going to validate this model, right? It's from single cell data. The key is really now to validate single cell data sets and computational models in the wet lab. So we asked, so how can we validate this trajectory here? So what we did is actually we used PDGFR-beta RTD tomato mice, so you can fade trace PDGFR-beta cells, which are all pericytes and fibroblasts. You can fade trace them, and then you can sacrifice at different time points after myocardial infarction, sort the TD tomato cells and perform single cell sequencing. So you have a time course data set, and then this time course data set can integrate with the human data to ask really whether this trajectory happens over time, right? So we did this here. You can see there's very similar clusters as in human. You have this SCARA5 non-activated fibroblasts. We have this periostine positive myofibroblasts. So now we compared the mouse to the human data, and interestingly, most populations, I mean, the mouse data, you can always play with clustering. There's like slightly more clusters. At the end, you always have to validate. You can also over cluster these human cells. But what you can see is that these fibroblast 3, these fibroblast 2, and these fibroblast 1 in human, they're really similar to certain populations in mouse, but this fibroblast 4 in human we don't find in mouse. It doesn't really matter, but what we did then is an annotation cross species. So we annotated the mouse data with the human gene expression signatures. You can see that here, and then looked at the time course, and what you can see is that fibroblast 1 stay more or less stable and even decrease, and fibroblast 2 really increase. So this really verifies or validates this trajectory that fibroblasts 2 are the myofibroblasts that increase over time in this time course data in the mouse. So, and now just one example, what you can do with the spatial data to identify novel mechanisms. So this is looking at heterogeneity of myeloid cells in the human data, quite some heterogeneity. And interestingly, what we found using these machine learning algorithms of the spatial data is that fibroblasts 2, and these are the myofibroblasts, always predicts the presence of SPP1 macrophages, so a subset of macrophages in space and vice versa. So when you can see this here, when you have myofibroblasts in space, and this is now using a tool called cell to location to map every cell population to space. When you have myofibroblasts in space in the spatial sequencing data in the same areas, you have this subset of macrophages in space. And now there's a lot of new papers recently coming out from Pfizer, for instance, from other companies, saying that these SPP1 macrophages seem to be very critical, not only in the heart and the kidney, but also in the lung and the liver. I think David Brenner also recently showed some data in the liver. And other labs call them SCAR-associated macrophages, but these are very similar macrophages. So these are really always in the same location. So we asked when these are in the same locations, that might probably talk to each other, that might have a role in disease. This is just some validation of this with in situ hybridization in a larger cohort. Don't want to get into detail. So Conrad and Gideon then teamed up to really, ask why do these SPP1 macrophages always fall in the same spatial locations as these myofibroblasts. And so when we reanalyzed some data from one of our collaborators, Nadia Rosenthal from Jack's lab, this is time course data of mouse myocardial infarction, single cell sequencing data. And what we found is actually that also these SPP1 macrophages are present in their data, and they basically have the highest score of ECM regulating genes. So this is a gene set where we summarized all genes that are known to regulate extracellular matrix formation. And then we built this trajectory that these SPP1 macrophages are derived from a subset of monocytes. This is a trajectory basically, and one interesting gene we found alongside this trajectory of monocyte to SPP1 macrophage differentiation is CXCL4. So what we did is actually, I haven't done it myself, but so Conrad and Gideon actually, they knocked out CXCL4, and you can see that here, when you knock out CXCL4, the scar size of the myocardial infarction is much smaller. It's really reduced. You can quantify this here really nicely. You can do echocardiography after myocardial infarction, so ultrasound. You can see the cardiac function is really rescued when you knock out CXCL4. And it's very similar in the kidney. So this is now ischemia reperfusion injury in the kidney. You can see there's much less fibrosis. When you knock out CXCL4, you can quantify this here. So what's then interestingly, when we did single nuclear RNA sequencing from these mouse kidneys, and this is sham, this is ischemia reperfusion injury of the kidneys, this is wild type mouse, this is CXCL4 mouse, so you can see these SPP1 macrophages are only present in injury in the wild type, but they are gone in the CXCL4 knockout. So to make a long story short, what we really could demonstrate is that CXCL4 from platelets basically drives polarization of these macrophages towards SPP1 macrophages, and these macrophages then activate myofibroblasts to drive fibrosis. So this cellular crosstalk we could only identify by looking at the spatial data, which is, I think, quite interesting. So I'm just going to skip this summary because I want to show you just very quickly 6 more slides on ADPKD, because I heard today, and I just added this in the car, that you guys are working on ADPKD. So ADPKD, as I don't have to really say, is one of the most common hereditary diseases in the world. It's the most common autosomal dominant disease in the kidney. So 10% of our dialysis patients actually have ADPKD. So what Jack actually did in my lab, and he graduated actually as a PhD student at the age of 67, which is quite impressive. So it was just his life dream to do a PhD, and he was a research technician for 35 years before. And he then, after his PhD, I think he did 6 months of a postdoc and then retired. But he was really an amazing guy. And so what he was actually interested in is whether he can get organoids from adult human kidney. So, and we studied a lot of different cell populations. As it turned out, these CD24 cells that have been studied by Paolo Romagnani for many years, they can really build they can really grow organoids in the dish much better than any other cell types. And we developed a protocol to grow these adult organoids, are called tubuloids. It's a 4-phase protocol, and culture them for almost a year. And so then we actually, we characterize them. You have really functional proximal tubules in the dish. You have apical basolateral orientation. You have brush borders here. You have actually acetylated tubulin towards the lumen, which I think is very critical, particularly when you grow cysts, because you have all seen these cysts from iPSC, and their primary cilium is outwards. It's different from human disease. So what we then actually asked is whether we can induce cyst formation. So we did a lot of different protocols. This is a time-lapse video now. We did like a multiplex CRISPR-Cas gene editing of PKD1 or PKD2. And you can see there's actually a cyst now growing after PKD gene editing. There's already some cyst presence here. There's one cyst growing here. So when you gene edit the PKD1 or the PKD2 gene, which is basically the gene that causes the human disease, you get actually cyst formation in the dish. So we're really excited about this, and you can see this nicely here. This is a lentivirus for the CRISPR gene editing that also has a GFP, so you can actually get really GFP positive cysts. This is really what you get in the human disease. So we're using this now a lot to screen compounds to identify whether compounds reduce cyst formation, because the disease is really annoying. You have, I mentioned this, 10% of our dialysis patients. You have young patients that are 16 years old and have family history. You do ultrasound, you know they have the disease. On average, depending on the mutation, their kidneys fail when they are mid-50s to mid-70s, and there's not much you can do. There's tolvaptan. I show you this is one drug that is approved, but the issue with tolvaptan is that It has some toxicity to other organs, but the major issue is really you have to pee 7 liters per day. So depending on your patient, when the patient is bus driver or pilot, you can't really sell this drug, right? I mean, you can't run to the toilet every 30 minutes. So there's a lot of patients that just say, I don't want to risk my quality of life to take this drug now for the next 30 years to then win maybe 3-4 years to be dialysis free. So we did some single nuclear RNA sequencing in human ADPKD kidneys and also in these tubuloids with gene editing, and really showed there's some disease-driving pathways, and I don't want to get into the details here, but some of the disease-driving pathways in human are also really active in these gene-edited tubuloids. And then interestingly, what you can see, this only approved therapy, this tolvaptan, really reduces also cyst size in this model. So this really shows that the model is really similar to the human disease. And interestingly, when you use iPSC organoids, you can also grow really beautiful cysts. Benjamin Friedman does this a lot, and we collaborate with him here. But interestingly, these are not responsive to tolvaptan at all. And the reason for that is that the target of tolvaptan, this is the AVPR2 receptor, it's not expressed in any of these published single-cell data sets of iPSC organoids, but it's really expressed in these adult kidney tubuloids. So the target, which tells us basically that I think depending on which disease you study, so if you study tubulopathies probably, I think it might make a lot of sense to use these adult tubuloids, because they really represent a more mature cell state of these epithelial cells compared to induced pluripotent stem cells. So thank you very much, and particularly all the lab members, the collaborators we have, and the funding agencies, and I'm happy to take questions.
Thank you, Rafa, for this talk. Question from the floor.
Very nice thought. The SPP1 macrophages that you mentioned, are they part of M1 or M2 macrophages, or is it like totally separate type of macrophages?
I mean, they are somewhere in between, like when you talk now to these hardcore macrophage people, they say you shouldn't say M1 or M2 because it doesn't exist. There's a lot of states in between. It looks like when you look at signatures from, people that have published M1 versus M2, it's somewhere closer to M2, but not exactly like M2.
Is it just in the heart, or you see in the kidney or other places, let's say a skin wound?
No, SPP1 macrophages, so other people have also discovered them now recently and called them scar-associated macrophages. There's a Nature Immunology review on this. David Brenner has found them in the liver. There's a lung study. There's a Pfizer paper recently that has reported this. So it seems like these scar-associated or SPP1 macrophages, we just called them SPP1. SPP1 is a gene that encodes for osteopontin. It's not really specific for these cells in general, but it's specific expressed when you just look among myeloid cells. That's what we call them, SPP1. But I think scar-associated macrophages might be the better term, and they're really present in almost every organ in spatial relation to matrix and myofibroblasts.
I'm confused. The SPP1 is important for translocation or is pathogenic? The other way, if you knock it out.
No, we don't care about SPP1. I just mentioned this SPP1. SPP1 is just a marker gene, so we don't care. I mean, there's like Andreas Ehrlich had this paper that osteopontin, you probably saw it in Science Advances, it's secreted from epithelial cells and then acts on the lung. We don't see any pathophysiology in this model. It's just we use it as a marker gene. We found this one chemokine 604. There's some other mechanisms. This is secreted by these macrophages basically and drives their polarization. And then these macrophages with other signaling pathways activate fibroblasts.
Can we share?
Yeah.
So, do you think that they are acquiring a memory, such as an epigenetic memory, to somehow, because they are especially there, and you showed convincingly in the heart, you said also in other tissues, diseased tissue, I assume, that they are specifically linked to a kind of reaction. So, but this, what happens? They still are there after a while, and would these macrophages be able to recognize quicker worsening likely the evolution of the disease?
Yeah, very good question. So we haven't really studied this. I think that's a good question. So at the end, to really convincingly show that they acquire a memory, I think you probably have to take them out, maybe transplant them, and show that they're actually still present, right?
Can partially show this.
Yeah, I mean, but still, I mean, in the ATAC sequencing would just be a snapshot, then it wouldn't show that they can react different on a future injury stimuli, right?
Right, although you could at least compare with the epigenome data of other macrophages, for example, in sepsis, where memory has been proven.
Yeah.
And then you could.
No, that's true.
Consider.
That's a good idea.
Dan.
Thank you for the presentation. I have two technical questions. How many cells have you analyzed in single cell or single nuclei RNA sequencing? How many?
For this paper?
Yes.
Oh, I can't remember. It's something like a couple 100,000, 200,000 or something. I can't remember exactly. I mean, now we are moving towards really higher patient number, higher cell numbers, because we've seen there's so much heterogeneity in humans that sometimes the inter-individual differences are stronger than the disease versus healthy differences. So that's why we now develop technologies to scale up. I mean, it's very costly, but we do develop technologies to scale up with reducing cost, you know, that we multiplex cells by genetic variability, by SNPs, for instance. We have a paper that's coming out soon, hopefully, with a method where you can scale up and then hopefully do around 1 million cells in a single experiment.
And have you confirmed the single cell RNA sequencing results with special transcriptomics? I mean, the results obtained with special transcriptomics are the same of single cell RNA sequencing. Have you confirmed that special transcriptomics confirm the single cell RNA sequence?
You can't, because the spatial data is basically high, you know, lower resolution. So there's always several cells in a given spot, right? And they can be, if you have injury and there's immune cells, lymphocytes are quite small, you can have up to 20 cells in a given spot. So you can't confirm it, but you see different signatures in different space. And I think it's like having both data sets. You can use the ATAC sequencing data. So you can just look at accessibility of promoter regions, and then from this, estimate gene expression from the ATAC sequencing data. So there you can validate your single cell with the current single cell. With this single cell Visium pipeline, you can't. There are some new technologies. This is really the fastest moving field currently, like spatial transcriptomics. There's a bunch of different technologies, you know, seqfish, merfish. There's in situ sequencing, like the Xenium device. There's other technologies like DebitSeq. There's StereoSeq from VGI, and there's some technologies at the subcellular resolution that really allow you to also validate some of the single cell data.
I would just make a comment. I mean, you made really the things very, very easy. You know, you say we did this, we select this, but what is behind? How many hours of bioinformatics work and data analyzing? Because actually we have lab time that is quite reducing, but then that analysis that is that kills 7%.
No, I agree. I mean, it seems like, I mean, we realized early on that data generation is sometimes much easier than analysis, right? And it takes much longer to analyze this data. So, I mean, we had a large team. We had like, you know, 6, 7 computational people that worked on the data for almost 2 years to really get everything out. And it's a large team, and we really moved towards team science. We collaborated a lot with other groups, you know, for years in a large team where we have, you know, probably around 10 people. And also for the revision, then we have a lot of, you know, staff scientists in the lab, experts in different technologies to validate findings, because this can't be done by one person.
I mean, how this large database, public database, can help in this? Can you imagine a database of trajectory? You know what I mean?
No, I mean, I think what we are trying to build now really a lot is to integrate the data and build. So what we are trying to build, and we founded a company in this regard, we're trying to build like a knowledge graph on fibrosis, where we integrate all open source data, and we build some specific data sets from our patients and larger cohorts to really understand whether we find novel druggable targets in this data. So we say, okay, we can look at a target, we can look at whether there's freedom to operate, is there patents out there, is there other papers that confirm it, what is upstream, what is downstream on the target, is this druggable, is there a protein structure, is there an AlphaFold model, is this does this target have a pocket for a small molecule, would this be rather extracellular to be accessible for an antibody? So to have all these, together, which is also major effort, but then we hope we can actually use this data to predict what could be an interesting target, and then we validate this, but then I have a higher chance that the validation in the wet lab works, because historically we just have looked at Excel sheets, and in the Excel sheets that sorted the Excel sheet for full change of p-value, and then you look at a few hundred genes and you don't know, okay, what is the most interesting gene? You drink a lot of coffee, you Google it, but then you just find some, and then you get tired, and then you validate one where you have randomly found that it might be interesting. And so we are now trying to really have a data-driven approach that gives you all data that is out there at your fingertips to say this could be a really cool target. Can we really hit this target? And we're trying to do this similarly in the wet lab, so that you can also use an in silico perturbation tool. So you can have your data, you can ask computationally whether you knock out this gene, what happens to your other genes, what happens to your gene regulatory network, and then based on this finding you can design your validation experiment, which helps you really to reduce efforts in your wet lab, because you can, if you have 100 new targets, you can validate them all, but then you can use these computational tools to make it much more likely that your wet lab work works, and then you can validate 3, 4, and maybe 3 or 2 of these 4 will work.
And you say that anyway, these new technologies, make really, they cannot beat the human heterogene. So what do you think to apply this technology to rare disease, monogenic disease? Do they will help much more than diabetics or other technologies?
Yeah, I hope so. I think my feeling is so now I've moved. Initially when I started my lab, we did a lot of fate tracing, modeling in mouse models, and so now what we're trying to do, start with human data and then do also a data-driven approach. So we are trying also to build a database of mouse models. We're trying to build a database of organoid models, and we want to have a data-driven approach to select the model to validate our mechanisms, to say, okay, this is Alport disease or whatever other disease. This is actually the model, the time course where the mechanism in mouse or in this organoid model is mostly active. You can really select this. So I think for all kind of questions, you have to really think about, you can integrate, for instance, DNA sequencing in your pipeline. So to understand which cells have the mutation you're interested in or whatever. I think there's a lot of I think approaches in the field is really moving towards a data-driven, unbiased approach, and I like this. I've worked a lot in my career also on specific pathways like the hedgehog pathway, but I got bored just working on one pathway, and so now having this data is cool, because you have so many new ideas. You can't follow all of them, but you always have new ideas. You look at new stuff, you look at pathways you have never looked, you find genes you've never looked before, and I think this is what excites me about science, because you always get constantly new ideas based on your data.
The New England Journal of Medicine paper by Torres about the efficacy of the clinical, about marketing, has been criticized a lot, because in order to get a very little significant, it use 2,000 patients, because the variability is so much in the you don't have only 2 genes, but probably more than 2 genes.
Yeah.
I don't know how many. So the question why in your tubuloid, not organoid, tubuloid, a completely different story. You have the lack. Because in your tubuloid that you use, say you have a mostly proximal tubular cell and non-distal tubular cell.
Yeah.
What do you think?
No, I mean, I don't really know. I mean, it's also the question we had, and also some of the reviewers had. I mean, it's mostly proximal, and you know, the ADPKD folks say it's mostly from distal, right? Which could be, but I mean, at the end, if you isolate, there's these wonderful studies where you have isolated the entire nephron and show they're from every part of the nephron, and it seems like these cells express ADPR2. I'm not sure. We haven't done enough experiments though, but we have done these tubuloids like 20 times and always did 20 times single cell sequencing to really confirm it's always proximal. I could imagine, because these CD24 cells are scattered throughout the nephron, that you know, depending on your culture conditions and on the patient you isolate these from, that you have more proximal, more distal parts, right? So, you know, this is like something we don't know. And you know, the Toverplan, I agree. I mean, we saw this effect, which was really interesting, but I agree that it's not a good, you know, treatment we have to do better for these patients.
And I have another particular question. When you isolate a tubuloid, obviously you can have a tubuloid in which you have the prevalence of the proximal or the distal. But what about the external review? Because you know, proximal tubular cells, they never see salt or tonicity of the seaside.
No, I agree.
While the distal tubular cell, they always say tonicity is well high. So what reason you use tubuloids?
No, so I think you can actually, so when you isolate these tubuloids fresh from patients and sequence them earlier, let's say 3 weeks after isolation, you have some proximal, but you also have some distal parts, like DCT. But interestingly, when you then culture them over time using our protocol, somehow only the proximal cells proliferate. I could imagine if you change growth factors, if you change stimuli, then probably the distal cells proliferate more. That's something we have to see. But I would think these are also interesting to study other tubular diseases, and we're also trying to add primary fibroblasts to see whether we can, use these as a tool to basically study epithelial fibroblast crosstalk. You know, I think this could be another interesting approach. I mean, we also use a lot of iPSC-derived organoids, but I think for some questions, maybe having these primary cells might be better.
If there are no other questions, I think we can close this section again. And thanks to Rafael for.
Thank you.
Di attenzione, prima di salutarci abbiamo rimandato il coffee break che adesso si svolgerà nella sala.
So we have a coffee break. We are closing, Mr. President, we are closing the section and devo dirti che è stata una sessione, credo, bella, no? Forse bellissima, certamente interessante, forse un po' corto, forse un po' lungo, dipende dai gusti, ma la cosa che è venuta fuori al meglio di tutti, proprio eclatante, che non è una stata 3 talk diretti al nephrology, no, sono diretti 3 talk che erano diretti a chi ha un po' di curiosità, per cui le domande più belle, scusatemi con la parte, sono venute dai nostri colleghi oncologi, sono venuti da persone che non avevano niente a che fare con la cosa nefrologia, che dimostra che No, lasciamo stare questa cosa, che dimostra che la scienza è unica. Quando si pongono delle problematiche, le problematiche interessano tutti e quindi a questo punto cose di questo genere devono essere ripetute. Poi devo iniziare, I must thank you very much for the outstanding contribution that you have done, both of you, okay? And I hope that the stimuli that we have received, it's also get also true for you, that you have understand that here the people of Biogem is really interested when we do science, but especially when we do good science, and you are just an example of how good science can be done. Grazie.